Hey everyone, picture this. You're standing in a museum staring at a massive Tyrannosaurus rex skeleton, and all you can see is this terrifying, scaly reptilian monster straight out of your worst nightmares. But what if I told you that this iconic predator might have actually looked more like a giant feathered chicken than the lizard-like beast you're imagining right now? What if nearly everything you thought you knew about how dinosaurs looked was completely wrong? For over a century, we've been picturing dinosaurs as these cold-blooded, scaly creatures lumbering through prehistoric swamps. Hollywood reinforced this image with every monster movie, every documentary, every children's book. But science has a funny way of turning our assumptions upside down, and the story of feathered dinosaurs is one of the most revolutionary discoveries in paleontology. It's a tale that begins with a single controversial fossil and ends with a complete transformation of how we see these ancient giants. Before we dive into this mind-bending journey, make sure to hit that subscribe button and ring the notification bell, because trust me, you're going to want to catch every episode in this series. We're about to explore discoveries so groundbreaking that they've rewritten textbooks and forced museums around the world to completely redesign their exhibits. And stick around until the end, because I've got something special planned that will blow your mind about what we're still discovering today. Now, let me take you back to the mid-1990s, when paleontologists thought they had dinosaurs all figured out. The scientific community was comfortable with their understanding of these ancient reptiles. Sure, there were debates about whether they were warm-blooded or cold-blooded, but one thing seemed certain. Dinosaurs were scaly, just like their living relatives, crocodiles and lizards. That comfortable certainty was about to be shattered by a discovery in northeastern China that would change everything. In the Liaoning province of China, something extraordinary was happening. Fossil hunters were pulling specimens out of the ground that preserved details scientists had only dreamed of seeing. These weren't your typical dinosaur fossils, just bones embedded in rock. These were complete animals with soft tissues, internal organs, and most shocking of all, what appeared to be fur or feathers covering their bodies. The geological conditions in this region, during the early Cretaceous period, roughly 125 million years ago, were perfect for this kind of preservation. Volcanic ash deposits created fine-grained sediments that could capture the most delicate details, while the lack of oxygen in ancient lake beds prevented decay. Then came the fossil that changed everything. Sinosauropteryx, a small theropod dinosaur about the size of a chicken, emerged from these Chinese fossil beds covered in what looked like a thick coat of dark filaments. When paleontologists first laid eyes on this specimen, many simply couldn't believe what they were seeing. Here was undeniable evidence that a dinosaur, a real non-avian dinosaur, had been covered in some kind of fuzzy, hair-like structures. The implications were staggering, and the scientific community erupted in heated debate. These weren't modern bird feathers as we know them today. Instead, they were simple, hollow filaments that resembled down, or even mammalian fur more than the complex flight feathers we see on birds. They were straight, evidently stiff, and composed of keratin and other proteins. Some of them even appeared to have been branched, suggesting a level of complexity that no one had anticipated in such ancient creatures. The discovery was so revolutionary that many paleontologists initially refused to accept it, clinging to the traditional view of scaly reptilian dinosaurs. But Sinosauropteryx was just the beginning. As more fossils emerged from the Liaoning deposits, it became clear that feathered dinosaurs weren't an anomaly. They were the norm. Species after species was discovered with various types of feather-like integument, each one adding another piece to an increasingly complex puzzle. Some had simple filaments like Sinosauropteryx. Others possessed more elaborate structures that resembled the downy feathers of baby birds. Still others displayed complex branched feathers with central shafts and intricate arrangements of barbs. The diversity was absolutely breathtaking. Scientists found dinosaurs with hollow quills similar to those of porcupines, massive tail fans like those of peacocks, and even the familiar pennaceous feathers we see on modern chickens. All of this in creatures that lived millions of years before the first true birds took to the skies. The traditional image of dinosaurs as uniformly scaly reptiles was crumbling with every new discovery, replaced by a vibrant, feathered world that was more alien and wonderful than anyone had imagined. What made these discoveries even more remarkable was their preservation quality. The Liaoning fossils didn't just show that feathers were present. They revealed how these feathers were arranged on the body, their colours, and even their molecular composition. Advanced analytical techniques allowed scientists to identify the same beta proteins that make up modern bird feathers, confirming that these ancient structures were indeed true feathers, not just superficially similar filaments. The geographic scope of these discoveries began to expand as well. While northeastern China remained the epicenter of feathered dinosaur research, important specimens began turning up in other locations around the world. Amber deposits from France preserve three-dimensional feathers in exquisite detail, showing transitional forms between simple filaments and complex flight feathers. Spanish limestone revealed carbonized imprints of branched filaments, while Slovenian rocks contained evidence of pennaceous feathers with sophisticated aerodynamic properties. Each new discovery raised profound questions about dinosaur biology and evolution. If feathers were present in so many different species across such vast spans of time and geography, when did they first evolve? How widespread were they among dinosaur groups? And perhaps most intriguingly, what were they originally used for? The answers to these questions would prove to be even more surprising than the discovery of feathers themselves. The evolutionary implications began to sink in as paleontologists studied the distribution of feathered species. Feathers weren't just present in the theropod dinosaurs that eventually gave rise to birds. They were turning up in ornithischians, a completely separate lineage of plant-eating dinosaurs that included species like Triceratops and Stegosaurus. This distribution suggested something truly remarkable. Feathers might have been present in the common ancestor of all dinosaurs, making them a fundamental feature of dinosaur biology, rather than a late evolutionary innovation. The discovery of feathered ornithischians was particularly shocking, because these dinosaurs were as distantly related to birds as crocodiles are to mammals. Finding feathers in these creatures was like discovering that elephants and tigers both had fur independently. It suggested that the capacity for feather development was deeply embedded in the dinosaur genetic toolkit, waiting to be expressed in different ways across millions of years of evolution. Modern molecular analysis has provided even more stunning insights into these ancient feathers. By designing antibodies that bind to specific keratin proteins, scientists have been able to identify the exact chemical composition of fossilized feathers from dinosaurs that lived between 160 and 75 million years ago. These studies have revealed a complex evolutionary transition in feather chemistry that coincided with the development of flight capabilities. The evolution of specialized beta-keratins appears to have been crucial for creating the lightweight yet strong structures necessary for powered flight, representing a key innovation in the path from ground-dwelling dinosaurs to aerial birds. The evidence for feathers extends beyond direct fossil impressions to include subtle anatomical clues that strongly suggest their presence, even in species where soft tissue preservation wasn't optimal. Quill knobs, those small bony bumps that serve as attachment points for large feathers in modern birds, have been discovered in the arm bones of famous dinosaurs like Velociraptor. These specialized structures would have been completely unnecessary without substantial feathered appendages, providing compelling indirect evidence for feathers in some of paleontology's most iconic species. Even more intriguing are the pygostyles found in various dinosaur species. These are collections of fused tail vertebrae that in modern birds provide support for elaborate tail plumage. Their presence in dinosaurs suggests that these ancient creatures possessed complex tail displays, similar to those of peacocks or turkeys, indicating sophisticated behavioral repertoires that we're only beginning to understand. The story becomes even more fascinating when we consider the functional significance of these feathers. While our first instinct might be to assume they were used for flight, the evidence suggests a much more complex picture. Many of the most elaborately feathered dinosaurs were clearly flightless, with body plans completely unsuited for aerial locomotion. Their feathers served other purposes entirely, functions that may have been even more important for their survival and reproduction than the ability to fly. The preservation of actual feather colours in some fossils has provided unprecedented insights into dinosaur behaviour and ecology. By analysing preserved melanosomes, the cellular structures that contain pigments, scientists have reconstructed the original colours of dinosaur plumage. Some species possessed brilliantly coloured feathers with iridescent blacks, rich browns, and possibly even structural colours that would have created blue and green hues, similar to those seen in modern peacocks and hummingbirds. This evidence for colourful plumage suggests that many dinosaurs engaged in elaborate visual displays, using their feathers for communication, species recognition, and sexual selection. The complexity and variety of these colour patterns indicate sophisticated social behaviours and mating systems that rival those of modern birds. Dinosaurs weren't just surviving in their ancient world. They were thriving, communicating, and competing in ways that required complex behavioural adaptations and social intelligence. The technological revolution in paleontology has been crucial to these discoveries. Scanning electron microscopy reveals microscopic details of feather structure that would be invisible to the naked eye. Synchrotron radiation allows scientists to peer inside fossils without damaging them, revealing hidden structures within the rock matrix. Mass spectrometry can identify organic compounds preserved for millions of years, providing direct chemical evidence of feather composition and coloration. These advanced techniques continue to push the boundaries of what we can learn from fossils, with new discoveries emerging regularly as technology improves. The integration of multiple analytical approaches provides a comprehensive understanding of feathered dinosaurs that goes far beyond what traditional paleontological methods could achieve alone. We're not just finding more feathered dinosaurs, we're understanding them in ways that would have seemed like science fiction just a few decades ago. But perhaps the most profound implication of feathered dinosaurs is what they tell us about the nature of evolutionary change itself. The transition from scales to feathers wasn't a sudden dramatic leap. Instead, it was a gradual process involving multiple intermediate stages, each with its own adaptive advantages. Recent discoveries have shown that this transition didn't happen uniformly across the body, but occurred in distinct zones, with some areas developing feathers while others retained reptilian scales. This zoned development pattern provides crucial insights into how major evolutionary innovations arise. Rather than wholesale transformations, evolution often works through gradual modifications of existing structures, with new functions emerging as byproducts of changes that originally served entirely different purposes. The story of feathered dinosaurs is ultimately the story of how life finds new solutions to ancient problems, creating beauty and complexity through the simple process of natural selection, acting over vast spans of time. Spans of time. The puzzle of dinosaur feathers becomes even more intricate when we examine the timeline of their evolution and the sheer variety of forms they took. Scientists have identified at least 5 distinct stages of feather development, each representing a major evolutionary leap from simple filaments to the sophisticated flight apparatus we see in modern birds. This wasn't a linear progression happening in a single lineage, but rather a complex branching pattern, where different dinosaur groups independently developed various feather types for completely different purposes. The earliest stage involved those simple, hollow filaments we saw in Sinosauropteryx, basically prehistoric fuzz that provided basic insulation. But even these primitive structures represented a massive innovation in vertebrate biology. The ability to produce keratin-based filaments required significant changes in skin development and protein production. changes that would have ripple effects throughout dinosaur physiology. These early proto-feathers were revolutionary not because they enabled flight, but because they allowed dinosaurs to regulate their body temperature more effectively than any reptile before them. Stage 2 brought branching filaments, where single strands split into multiple branches like tiny organic trees. This development dramatically increased the surface area available for insulation while maintaining the lightweight properties that made feathers so advantageous. Some dinosaurs stopped here, content with their fuzzy coats that kept them warm during cool nights and helped them maintain optimal body temperatures during the day. The third stage introduced the central shaft, or rachis, that would become the backbone of all advanced feather types. This innovation allowed for much larger, more structured feathers that could serve purposes beyond simple thermoregulation. With a central support structure, feathers could become display organs, communication tools, and eventually the foundation for powered flight. The development of the rachis represents one of those pivotal moments in evolutionary history, where a seemingly minor structural change opens up entirely new possibilities. Stages 4 and 5 brought the complex pennaceous feathers we recognize today, complete with barbs, barbules, and the intricate hook and groove system that allows individual feather elements to zip together into coherent flight surfaces. But here's where the story gets really interesting. Many dinosaurs that possessed these advanced feather types never flew a day in their lives. They were using flight-ready equipment for entirely terrestrial purposes, suggesting that the evolution of flight was more of an opportunistic co-option of existing structures rather than a directed march toward aerial capability. The behavioral implications of these discoveries are staggering. Imagine walking through a Cretaceous forest and encountering a pack of velociraptors, but instead of the scaly monsters from Hollywood films, you see creatures that look more like deadly, flightless birds. Their arms would have been covered in long, asymmetrical feathers, similar to those of modern flying birds. But these raptors used them for display, intimidation, and possibly even to help corral prey. When they spread their feathered arms wide, they would have created impressive visual barriers that could confuse and direct the movements of their victims. The evidence for complex social behaviors among feathered dinosaurs continues to mount with each new discovery. Some specimens have been found in brooding positions, with their feathered arms spread protectively over nests full of eggs. This behavior, identical to that seen in modern birds, suggests that parental care was sophisticated and that feathers played a crucial role in protecting and incubating the next generation. The fact that we can observe these behaviors preserved in stone speaks to the exceptional preservation conditions that captured these intimate moments from deep time. Color analysis has revealed that dinosaur feathers weren't just functional, they were spectacular. Some species possessed iridescent plumage that would have shimmered with metallic sheens in sunlight, creating displays that rivaled the most colorful birds alive today. Others had bold patterns of stripes, spots, and solid colors that served as species identification markers and sexual advertisements. The discovery that a small dinosaur called Anchiornis was primarily black with white wing stripes and a reddish crest gives us our first glimpse into the actual appearance of these ancient creatures, and it's far more vibrant and beautiful than anyone had imagined. The preservation of these colors in fossils represents one of the most remarkable achievements in paleontology. The melanosomes responsible for producing these hues are incredibly tiny, just nanometers across, yet they've survived for over 100 million years in some specimens. Different shapes of melanosomes produce different colors, and by comparing fossil melanosomes to those in modern birds, scientists can reconstruct the exact shades and patterns that adorned dinosaur feathers. This level of detail was unimaginable just a few decades ago, yet it's becoming routine in modern paleontological research. The geographic distribution of feathered dinosaur discoveries tells its own fascinating story. While China's Liaoning province remains the crown jewel of feathered dinosaur sites, important specimens have emerged from deposits around the world. Each locality provides unique insights into different aspects of feather evolution and dinosaur biology. The amber deposits of Myanmar have preserved feathers in three-dimensional detail that allow scientists to study their microstructure, as if they were examining modern specimens under a microscope. European discoveries have been particularly important for understanding the early stages of feather evolution. Spanish amber has yielded primitive feathers that show the transition from simple filaments to more complex branched structures, providing crucial missing links in our understanding of how feathers evolved. These specimens are often small and fragmentary, but they fill in gaps in the evolutionary sequence that help us understand how major innovations arise through gradual modification of existing structures. The molecular revolution in paleontology has added another dimension to feathered dinosaur research. Scientists can now extract and analyze proteins from fossils that are over 100 million years old, providing direct chemical evidence of feather composition and evolution. These studies have revealed that the proteins making up dinosaur feathers underwent significant changes over time, becoming lighter and more flexible as flight capabilities evolved. This molecular evidence perfectly complements the morphological data from fossil feathers, creating a comprehensive picture of how these structures changed through deep time. Advanced imaging techniques have revolutionized how we study these fossils. Synchrotron radiation can reveal details invisible to conventional methods, showing the internal structure of fossilized feathers and even identifying previously unknown specimens within rock samples. CT scanning allows researchers to peer inside specimens without damaging them, revealing three-dimensional arrangements of feathers and bones that provide insights into how these structures functioned in life. The discovery of quill knobs in velociraptor arms sent shockwaves through the paleontological community. These small bumps on the forearm bones serve as attachment points for large feathers in modern birds, and their presence in a dinosaur that lived 75 million years ago provided undeniable evidence that one of paleontology's most famous predators was feathered. The implications were profound, not just for our understanding of Velociraptor, but for the entire family of raptor dinosaurs. If Velociraptor had large feathers, it's likely that most or all of its relatives did as well. The function of these feathers in raptors remains a subject of intense scientific debate. They clearly weren't used for flight, as these animals were far too heavy and lacked the necessary skeletal modifications for powered flight. Instead, they likely served multiple purposes, including display, thermoregulation, and possibly even prey manipulation. Some scientists have suggested that raptors used their feathered arms like nets, spreading them wide to prevent prey from escaping, while the pack closed in for the kill. Pygostyles, those fused tail vertebrae that support elaborate tail feathers in modern birds, have been discovered in numerous dinosaur species. The presence of these structures indicates that many dinosaurs possessed complex tail displays, similar to those of peacocks or turkeys. Imagine an oviraptor spreading its magnificent tail fan in a prehistoric mating display, its iridescent feathers catching the light as it performed an elaborate courtship dance. These behaviors, long thought to be uniquely avian, were apparently common among dinosaurs, tens of millions of years before the first true birds evolved. The story of feathered dinosaurs is still being written, with new discoveries emerging at a rapid pace. Recent finds have pushed the origins of feathers even deeper into the past, with some evidence suggesting that primitive feather-like structures may have been present in the common ancestor of all archosaurs, the group that includes dinosaurs, birds, crocodiles, and their extinct relatives. If true, this would mean that feathers are an incredibly ancient innovation, one that predates the dinosaurs themselves by tens of millions of years. The implications for our understanding of dinosaur physiology are equally profound. Feathers are expensive structures to produce and maintain, requiring significant energy investment and sophisticated developmental programs. Their widespread presence among dinosaurs suggests that these animals had metabolic rates more similar to modern birds and mammals than to contemporary reptiles. This evidence supports the growing consensus that dinosaurs were active, warm-blooded creatures with complex behaviors and social structures. The preservation of feathers in amber has opened up entirely new avenues of research. These three-dimensional specimens retain details that are lost in compression fossils, allowing scientists to study the microscopic structure of prehistoric feathers with unprecedented clarity. Some amber specimens even preserve the barbules and hooklets that allow feather barbs to zip together, providing direct evidence of the sophisticated engineering that makes bird flight possible. The discovery of feathered dinosaurs has forced a complete re-evaluation of dinosaur ecology and behavior. These weren't the sluggish, cold-blooded monsters of popular imagination, but rather active, colourful creatures that engaged in complex social interactions. They courted mates with elaborate displays, cared for their young with sophisticated parental behaviours, and communicated through visual signals that we're only beginning to understand. The Mesozoic world was far more vibrant and dynamic than anyone had previously imagined. Modern reconstructions of feathered dinosaurs represent a complete departure from traditional depictions. Museums around the world have been forced to update their exhibits, replacing scaly reptilian monsters with creatures that look more like bizarre, flightless birds. These new reconstructions are based on solid scientific evidence, incorporating data on feather structure, coloration, and arrangement that allows artists to create accurate depictions of how these animals actually appeared in life. The technological advances driving these discoveries continue to accelerate. New analytical techniques are being developed that can extract even more information from fossils, including details about feather growth patterns, seasonal molting cycles, and even stress indicators that reveal aspects of dinosaur behavior and ecology. The field of paleontology is experiencing a golden age of discovery, with feathered dinosaurs leading the way into new frontiers of ancient life reconstruction. The story of how dinosaurs evolved feathers is ultimately a story about innovation and adaptation. These structures didn't evolve in isolation, but rather as part of a complex suite of changes that transformed dinosaur biology and behavior. Feathers enabled new forms of communication, more efficient thermoregulation, enhanced parental care, and eventually, the conquest of the skies. They represent one of evolution's greatest success stories, an innovation so successful that it continues to dominate aerial ecosystems today in the form of modern birds. Birds. The revelation that feathers weren't just a bird innovation has fundamentally altered our understanding of what it means to be a dinosaur. When we examine the fossil record more closely, patterns emerge that suggest feathers may have been far more common than the scattered discoveries initially indicated. The problem isn't that feathered dinosaurs were rare, it's that the conditions necessary to preserve soft tissues like feathers are extraordinarily uncommon in the geological record. Think about it this way. For every feathered dinosaur fossil we've discovered, there were likely thousands of feathered species that lived and died without leaving any trace of their integument. Soft tissues decay rapidly after death, and only under very specific circumstances do they become fossilized. The volcanic lake beds of northeastern China created a perfect storm of preservation conditions that captured details normally lost to time. Fine volcanic ash settled quickly over carcasses, creating an oxygen-poor environment that prevented bacterial decay, while the mineral-rich water gradually replaced organic tissues with stone. This preservation bias means that our understanding of feathered dinosaurs is still heavily skewed toward a few exceptional localities. But as paleontologists have learned to recognize the subtle signs of feather attachment in bones, even fragmentary skeletons from around the world are beginning to tell stories of prehistoric plumage. The quill knobs found on velociraptor arm bones weren't initially recognized for what they were. It took decades of comparative anatomy research with modern birds before scientists realized these small bumps were smoking guns, undeniable evidence of substantial feather attachment even in fossils where no soft tissues survived. The chemical analysis of fossilized feathers has opened up an entirely new dimension of prehistoric research. When scientists first attempted to extract proteins from dinosaur fossils, many colleagues were skeptical that any organic molecules could survive for millions of years. The breakthrough came when researchers realized that the proteins in feathers are some of the most durable biological materials on Earth. Beta-keratins, the primary structural proteins in feathers, are incredibly stable and can persist in fossils under the right conditions for tens of millions of years. These protein analyses have revealed something remarkable about feather evolution. The earliest dinosaur feathers contained a mixture of protein types, some more similar to those found in scales, and others resembling modern bird feathers. Over time, there was a clear evolutionary trend toward lighter, more flexible proteins that would have made feathers better suited for aerodynamic functions. This molecular evidence provides a completely independent line of support for the morphological data showing increasing feather complexity through time. The discovery of melanosomes in fossilized feathers represents one of the most exciting developments in paleontology. These microscopic structures, responsible for producing colors in modern animals, have been preserved in exquisite detail in some dinosaur fossils. Different shapes and arrangements of melanosomes produce different colors, and by comparing fossil melanosomes to those in living birds, scientists can reconstruct the exact hues that adorn prehistoric creatures. The first dinosaur to have its colors scientifically reconstructed was Sinosauropteryx, that same small theropod that started the feathered dinosaur revolution. Analysis of its fossilized melanosomes revealed that this pioneering discovery was reddish-brown with darker stripes across its tail and back. This wasn't just academic curiosity. The color pattern suggested that Sinosauropteryx lived in forested environments where such camouflage would have been advantageous, providing insights into prehistoric ecology that go far beyond simple appearance. Subsequent color reconstructions have revealed a stunning diversity of dinosaur plumage. Anchiornis, a small feathered dinosaur from China, was primarily black with bold white stripes on its wings and a reddish crest on its head. Psittacosaurus, a plant-eating dinosaur with both scales and feathers, had complex camouflage patterns that would have helped it blend into prehistoric forest floors. These aren't artistic interpretations or educated guesses. They're scientific reconstructions based on direct analysis of preserved pigment structures. The implications of colorful dinosaur plumage extend far beyond aesthetics. Color serves multiple functions in modern animals, from camouflage and thermoregulation to communication and sexual selection. The elaborate patterns preserved in dinosaur fossils suggest that these ancient creatures engaged in complex visual communication, using their plumage to signal everything from species identity to reproductive readiness. The diversity of color patterns also indicates that dinosaurs occupied a wide variety of ecological niches, each requiring different camouflage and signaling strategies. Some of the most spectacular discoveries have come from amber deposits that preserve three-dimensional feathers in perfect detail. Unlike compression fossils that flatten specimens into 2 dimensions, amber specimens retain their original shape and structure. Scientists can examine these prehistoric feathers under powerful microscopes and observe details that would be impossible to see in traditional fossils. The barbules that allow feather veins to zip together, the microscopic hooks that create aerodynamic surfaces, even the hollow internal structure that makes feathers so lightweight. All of these features are preserved in amber specimens. The amber feathers also provide crucial evidence about the evolution of flight. Many specimens show transitional features between the simple filaments of early dinosaurs and the complex aerodynamic surfaces of modern birds. Some have asymmetrical veins that would have provided lift and thrust, while others show symmetrical patterns more suited for display or insulation. This diversity suggests that different dinosaur lineages were experimenting with various feather designs, some of which eventually led to powered flight, while others served entirely different functions. Recent discoveries have pushed the origins of feathers even deeper into the past. Fossil evidence suggests that primitive feather-like structures may have been present in pterosaurs, the flying reptiles that were cousins to dinosaurs. If confirmed, this would indicate that the capacity for feather development was present in the common ancestor of pterosaurs and dinosaurs, pushing the origin of these structures back by tens of millions of years. The study of feather development in modern birds has provided crucial insights into how these structures evolved in dinosaurs. Feathers don't just appear fully formed, they develop through a complex series of genetic and cellular processes that can be traced back through evolutionary time. By studying how feathers grow in bird embryos, scientists can understand the developmental mechanisms that would have been active in dinosaur skin. This research has revealed that the genetic toolkit for feather production is ancient and complex, involving dozens of genes that control everything from initial feather bud formation to the final shape and structure of mature feathers. The molecular machinery required for feather production represents a major evolutionary innovation. Unlike scales, which are relatively simple structures, feathers require sophisticated cellular mechanisms to produce their complex branched architecture. The evolution of these mechanisms would have been a gradual process, with each step providing some advantage that was preserved by natural selection. The intermediate stages preserved in the fossil record show exactly how this process unfolded over millions of years. The behavioral evidence for feathered dinosaurs continues to accumulate as more fossils are discovered and analyzed. Specimens have been found in brooding positions with their feathered arms spread over nests, suggesting that parental care was sophisticated and that feathers played a crucial role in protecting eggs and young. Other fossils show evidence of communal nesting, with multiple adults sharing responsibility for incubating eggs and caring for hatchlings. These behaviors are complex and require significant cognitive abilities, suggesting that feathered dinosaurs were far more intelligent than traditionally assumed. The discovery of feathers in herbivorous dinosaurs has been particularly surprising. These plant eaters had no need for the predatory adaptations that might explain feathers in carnivorous species, yet they too possessed elaborate plumage. In some cases, the feathers were concentrated around the tail and arms, creating impressive display structures that would have been used for communication and courtship. The presence of feathers in herbivorous dinosaurs strongly suggests that display and thermoregulation were the primary drivers of feather evolution, with flight capabilities emerging later as a secondary adaptation. The preservation of dinosaur skin alongside feathers has provided unique insights into how the transition from scales to feathers occurred. Rather than a wholesale replacement of one structure type with another, the evolution of feathers appears to have been a gradual zone-specific process. Some areas of the body developed feathers while others retained scales, creating a mosaic pattern that persisted for millions of years. This zoned development helps explain how such a major evolutionary transition could occur without disrupting essential skin functions like protection and moisture regulation. Modern techniques for studying fossil preservation have revealed that many more specimens contain traces of feathers than previously recognized. What were once dismissed as artifacts or mineral stains are now being identified as genuine feather impressions through careful chemical and microscopic analysis. This has led to the recognition of feathered integument in dinosaur species where it was previously unknown, expanding our understanding of how widespread these structures really were. The engineering principles underlying feather design continue to inspire modern technology. The lightweight yet strong construction of feathers has influenced everything from aircraft wing design to advanced composite materials. Understanding how dinosaurs first developed these remarkable structures provides insights into how natural selection can produce solutions to engineering problems that human technology is only beginning to appreciate. The study of feathered dinosaurs has also revolutionized our understanding of dinosaur metabolism and physiology. Feathers are metabolically expensive structures that require significant energy to produce and maintain. Their widespread presence among dinosaurs indicates that these animals had high metabolic rates, more similar to modern birds and mammals than to contemporary reptiles. This evidence supports the growing consensus The dinosaurs were active, warm-blooded creatures, capable of sustaining high levels of activity. The preservation of growth patterns in fossilized feathers has revealed that dinosaurs underwent regular molting cycles similar to modern birds. Some specimens show evidence of seasonal color changes, suggesting that dinosaur plumage varied throughout the year for camouflage or display purposes. These discoveries provide unprecedented insights into the annual life cycles and behaviors of creatures that lived over 100 million years ago. The technological revolution continues to accelerate the pace of discovery in feathered dinosaur research. New analytical techniques are being developed that can extract even more information from fossils, including details about feather chemistry, growth rates, and even stress indicators that reveal aspects of dinosaur health and behavior. Artificial intelligence is being used to analyze vast databases of fossil images, automatically identifying feather traces that might be missed by human observers. The global nature of feathered dinosaur discoveries is becoming increasingly apparent as researchers learn to recognize the subtle signs of feather preservation in fossils from around the world. Specimens from Mongolia, Argentina, and even Antarctica are yielding evidence of feathered integument, suggesting that these structures were truly global in their distribution during the Mesozoic era. Each new locality provides unique insights into regional variations in dinosaur plumage and the environmental factors that influenced feather evolution. The story of feathered dinosaurs represents more than just a scientific discovery. It's a testament to the power of evidence to overturn long-held assumptions and transform our understanding of the natural world. The scaly reptilian monsters of popular culture have been replaced by vibrant feathered creatures that were far more complex and beautiful than anyone had imagined. This transformation continues today, as new discoveries push the boundaries of what we know about these remarkable animals and their evolutionary legacy in the form of modern birds. The preservation of feather impressions in rock tells a story of geological precision that's almost miraculous in its rarity. When you consider the chain of events necessary to capture something as delicate as a feather in stone, it becomes clear why these discoveries are so precious and why they've taken so long to accumulate in the scientific literature. The process begins with rapid burial, typically in fine-grained sediments that can capture microscopic details. Oxygen must be excluded quickly to prevent bacterial decomposition, and the chemical conditions must be just right to allow mineralization of organic tissues without destroying their structure. The Jehol biota of northeastern China represents the gold standard for this type of preservation. But it's not the only window into the feathered world of prehistoric life. Similar conditions occurred sporadically throughout the Mesozoic era in various parts of the world, creating scattered snapshots of ancient ecosystems where soft tissues were preserved alongside bones. Each of these preservation events captured different aspects of feathered dinosaur biology, from the simple filaments of early species to the complex aerodynamic structures that would eventually enable powered flight. The chemical fingerprints left by ancient feathers have revealed surprising details about their composition and function. Mass spectrometry analysis of fossilized specimens has identified not just the primary structural proteins, but also traces of lipids and other organic compounds that provided waterproofing and flexibility. These chemical signatures indicate that dinosaur feathers possessed many of the same properties as modern bird feathers, including the ability to repel water and maintain their structure under stress. The evolution of feather microstructure represents one of the most remarkable examples of biological engineering in the fossil record. The hollow shaft design that characterizes advanced feathers provides maximum strength with minimum weight, a principle that modern aerospace engineers spend billions of dollars trying to replicate. The branching pattern of barbs and barbules creates a hierarchical structure that can repair itself when damaged, allowing individual feathers to maintain their function even after significant wear. This sophisticated architecture didn't emerge overnight, but evolved through millions of years of incremental improvements. Advanced imaging techniques have revealed that some dinosaur feathers possessed structural elements that were previously unknown in the fossil record. Scanning electron microscopy has identified microscopic hooks and grooves that allowed feather elements to interlock in complex patterns, creating surfaces with specific aerodynamic or display properties. These structural innovations represent evolutionary experiments that sometimes succeeded in producing new capabilities and sometimes led to dead ends that were abandoned by natural selection. The relationship between feather structure and function becomes particularly fascinating when examining species that clearly couldn't fly. Large theropods like Eutyrannus, a feathered relative of Tyrannosaurus rex that weighed over a ton, possessed elaborate plumage that served purposes entirely unrelated to aerial locomotion. These massive predators used their feathers for thermoregulation in cold climates, and possibly for intimidating rivals or prey. The image of a giant tyrannosaur covered in fuzzy feathers might seem comical, but it represents a sophisticated adaptation to environmental challenges. The discovery of asymmetrical feathers in flightless dinosaurs has provided crucial insights into the evolution of aerodynamic structures. These feathers, with their characteristic shape that generates lift in flying birds, appeared millions of years before their bearers could use them for flight. This represents a classic example of pre-adaptation, where structures evolve for one purpose and are later co-opted for entirely different functions. The presence of flight-ready feathers in ground-dwelling dinosaurs suggests that powered flight was almost inevitable once these structures reached sufficient complexity. Trace fossil evidence has begun to complement direct feather preservation in unexpected ways. Dinosaur trackways sometimes preserve impressions of feathers that brushed against soft sediment as the animals moved, providing evidence of plumage in species where body fossils haven't been found. These traces indicate that feathered dinosaurs were walking through ancient landscapes across the globe, leaving subtle signatures of their passage that paleontologists are only now learning to recognize and interpret. The study of feather attachment sites on dinosaur bones has revealed sophisticated muscular systems that controlled plumage positioning and movement. Just as modern birds can raise and lower their feathers for communication or thermoregulation, many dinosaurs possess the anatomical machinery necessary for complex feather control. This neuromuscular coordination would have required significant brain power, adding to the growing evidence that dinosaurs were far more intelligent and behaviorally complex than traditionally assumed. Recent discoveries have shown that feather evolution wasn't a simple linear progression from simple to complex. Different dinosaur lineages independently developed various feather types, sometimes converging on similar solutions to environmental challenges. This parallel evolution demonstrates the fundamental utility of feathered integument and explains why these structures became so widespread among dinosaur groups. The redundancy in feather evolution also suggests that the genetic mechanisms underlying feather development are robust and easily activated when environmental conditions favor their expression. The molecular clock data derived from ancient protein analysis has provided new insights into the timing of feather evolution. By comparing the rate of protein sequence changes in fossil feathers to those in modern birds, scientists can estimate when different feather innovations first appeared. These molecular dates often precede the oldest known fossils by millions of years, suggesting that feathered dinosaurs were far more ancient than the direct fossil evidence initially indicated. The preservation of feather growth patterns in fossils has revealed that dinosaurs underwent molting cycles similar to modern birds, but with some interesting differences. Some species appear to have molted gradually throughout the year, while others show evidence of seasonal molts that coincided with breeding cycles or migration patterns. The timing and pattern of these molts provide insights into dinosaur physiology and behavior that would be impossible to obtain from bones alone. The discovery of specialized feather types in different body regions has shown that dinosaur plumage was highly regionalized, with different areas serving distinct functions. Head crests often contain display feathers with vibrant colors and elaborate structures, while body feathers focused on insulation and camouflage. Wing feathers in some species were optimized for aerodynamic performance, even when the animals couldn't fly, suggesting that these structures may have served functions like prey herding or territorial displays. The chemical analysis of fossilized feather pigments has revealed that some dinosaurs possessed iridescent plumage that would have created stunning visual displays. These structural colors, produced by microscopic arrangements of keratin and air spaces rather than pigments alone, indicate a level of optical sophistication that rivals the most spectacular modern birds. The presence of such complex coloration systems suggests that dinosaur visual communication was far more elaborate than previously imagined. The study of feather development in dinosaur embryos has provided unique insights into how these structures formed during prehistoric growth. Fossilized eggs with developing feather buds show that dinosaur chicks began growing their plumage while still in the shell, emerging into the world already equipped with insulation and protection. This early feather development would have been crucial for temperature regulation and survival in the often harsh environments of the Mesozoic era. The biomechanical analysis of feathered dinosaur locomotion has revealed that plumage may have played important roles beyond display and thermoregulation. Computer models suggest that arm feathers could have provided significant aerodynamic assistance during running, allowing some species to achieve higher speeds or maintain stability while pursuing prey. The interaction between feathers and airflow during rapid locomotion represents a complex physics problem that scientists are only beginning to understand. The preservation of feather wear patterns in fossils has provided evidence of specific behaviors and environmental interactions. Some specimens show asymmetrical wear on wing feathers that suggests repeated contact with vegetation or substrate, indicating how these animals moved through their environments. Others display damage patterns consistent with aggressive interactions, providing direct evidence of combat or territorial disputes among feathered dinosaurs. The discovery of seasonal feather color changes in some dinosaur species has revealed sophisticated adaptations to environmental cycles. Melanosomes preserved in different growth bands of individual feathers show that some dinosaurs could alter their coloration throughout the year, possibly for camouflage during different seasons or for breeding displays. This level of physiological control over appearance indicates complex hormonal and developmental systems that were previously unknown in extinct species. The analysis of feather-bearing skin impressions has shown that the transition from scales to feathers occurred through intermediate structures that combined properties of both integument types. These transitional forms, preserved in exceptional detail in some specimens, provide crucial evidence for understanding how major evolutionary innovations arise through gradual modification of existing structures. The coexistence of scales and feathers on the same animal demonstrates that evolution often works through mosaic patterns rather than wholesale transformations. The study of feather fossilization processes has revealed that different preservation environments capture different aspects of these structures. While compression fossils preserve overall morphology and arrangement, amber specimens retain three-dimensional structure and microscopic detail. Permineralized specimens can preserve chemical composition and growth patterns, while trace fossils capture behavioral evidence. This diversity of preservation modes means that each new discovery has the potential to reveal previously unknown aspects of dinosaur biology. The technological advances driving feathered dinosaur research continue to accelerate at an unprecedented pace. Machine learning algorithms are being trained to recognize feather impressions in fossil photographs, potentially identifying specimens that have been overlooked in museum collections. Portable analytical equipment allows researchers to study fossils in the field, providing immediate feedback about preservation quality and scientific significance. These technological improvements are democratizing paleontological research and enabling discoveries that would have been impossible just a few years ago. The global distribution of feathered dinosaur discoveries continues to expand as researchers apply new techniques to fossils from around the world. Recent finds from South America, Africa, and Australia are filling in gaps in our understanding of how feathers evolved across different continents and climatic zones. Each new locality provides unique insights into regional variations in dinosaur plumage and the environmental factors that influenced feather evolution during the Mesozoic era. The story of feathered dinosaurs is fundamentally changing how we understand the relationship between form and function in evolutionary biology. These discoveries demonstrate that complex structures can evolve for one purpose and later be co-opted for entirely different functions, a process that has profound implications for understanding how major evolutionary innovations arise. The feathered dinosaur revolution continues to unfold with each new discovery, revealing a prehistoric world that was far more colourful, complex, and beautiful than anyone had previously imagined. Complex and beautiful than anyone had previously imagined. The integration of feathered dinosaur discoveries with our understanding of ancient ecosystems has painted a picture of Mesozoic environments that were far more dynamic and visually striking than anyone had previously conceived. When we reconstruct these ancient worlds based on the latest fossil evidence, we see landscapes populated by creatures that would have rivaled modern tropical rainforests in their diversity of colours, patterns, and behaviours. The forests of Cretaceous China weren't dominated by the drab, uniformly scaly reptiles of old museum dioramas, but rather by a spectacular array of feathered species, displaying everything from subtle earth tones to brilliant iridescent plumage. The ecological implications of widespread feather distribution among dinosaurs extend far beyond individual species appearance. Feathers fundamentally alter how animals interact with their environment, affecting everything from predator-prey relationships to mating systems and social hierarchies. A feathered predator moves differently through vegetation than a scaled one, potentially approaching prey more quietly due to the sound-dampening properties of plumage. Display behaviours become more complex when visual communication can incorporate not just body postures and movements, but also dynamic feather positioning and colour changes. Recent biomechanical studies have revealed that feathers may have served surprising functions in non-flying dinosaurs that we're only beginning to appreciate. Computer simulations of feathered theropod locomotion suggest that arm plumage could have provided significant aerodynamic benefits during high-speed pursuits, acting like natural spoilers that improved stability and maneuverability. Some species may have used their feathered arms as steering mechanisms while running, allowing them to make sharp turns that would have been impossible for purely terrestrial predators. The thermoregulatory advantages of feathers become particularly significant when we consider the climatic conditions that prevailed during peak dinosaur diversity. The Mesozoic era experienced dramatic temperature fluctuations, including periods of global warming and cooling that would have challenged the survival strategies of large vertebrates. Feathered dinosaurs possessed a distinct advantage in these changing conditions, able to adjust their body temperature through behavioral modifications like fluffing or compressing their plumage. This physiological flexibility may have been a key factor in their evolutionary success across diverse environments and time periods. The discovery of complex feather arrangements in dinosaur specimens has revealed sophisticated anatomical adaptations that parallel those found in modern birds. Specialized muscle groups controlled feather positioning, allowing dinosaurs to communicate through subtle plumage adjustments that would have been invisible to casual observers, but meaningful to members of their own species. These neuromuscular systems required significant evolutionary investment, suggesting that visual communication was critically important to dinosaur social structures. Isotope analysis of fossilized feathers has provided unprecedented insights into dinosaur migration patterns and seasonal behaviors. The chemical composition of feathers reflects the environment where they grew, creating a record of animal movements that can be read like a geological passport. Some dinosaur species show isotopic signatures indicating long-distance travel between different climatic zones, suggesting seasonal migrations that rival those of modern birds. This evidence for complex movement patterns adds another layer to our understanding of dinosaur behavioral sophistication. The preservation of parasites and other microorganisms within fossilized feathers has opened an entirely new field of prehistoric ecological research. These microscopic fossils reveal details about dinosaur health, disease resistance, and ecosystem interactions that would be impossible to study through bones alone. Some specimens contain evidence of feather lice and other parasites that provide insights into dinosaur grooming behaviors and social interactions. The presence of these organisms also indicates that dinosaur feathers supported complex microbial communities similar to those found on modern birds. Experimental archaeology has played an increasingly important role in understanding how dinosaur feathers functioned in ancient environments. Researchers have created physical models of dinosaur feathers based on fossil evidence and tested their aerodynamic, insulation, and display properties under controlled conditions. These experiments have revealed that even relatively simple feather structures could provide significant advantages in terms of temperature regulation and visual communication, explaining why these innovations spread so rapidly through dinosaur lineages. The study of feather wear patterns in fossil specimens has provided direct evidence of specific behaviors and environmental interactions that shaped dinosaur daily life. Asymmetrical wear on wing feathers suggests repeated contact with vegetation during foraging, while damage patterns on tail plumes indicate aggressive interactions during territorial disputes. Some specimens show evidence of deliberate feather maintenance, with wear patterns consistent with regular preening behaviors that would have been essential for maintaining plumage function. Advances in protein sequencing from ancient specimens have revealed that dinosaur feathers contained a diverse array of structural proteins, many of which have no direct equivalent in modern bird feathers. These unique protein compositions may have provided dinosaur plumage with properties that are no longer present in living species, such as enhanced durability or specialized optical characteristics. The loss of these protein variants during the transition to modern birds represents an example of evolutionary trade-offs where some capabilities were sacrificed in favor of others. The reconstruction of dinosaur social behaviors based on feather evidence has challenged many assumptions about reptilian intelligence and complexity. Species with elaborate display plumage clearly engaged in sophisticated courtship rituals that would have required significant cognitive abilities to execute effectively. The coordination necessary for group displays, where multiple individuals synchronize their movements and feather positions, suggests communication systems that approach the complexity of modern bird flocks. Chemical analysis of fossilized feathers has identified traces of pheromones and other chemical signals that dinosaurs may have used for communication. These organic compounds, preserved in microscopic quantities within feather structures, provide evidence for olfactory communication systems that complemented visual displays. The combination of chemical and visual signaling would have created rich communication networks that allowed dinosaurs to convey complex information about identity, reproductive status, and territorial boundaries. The discovery of seasonal molting patterns in dinosaur fossils has revealed that these animals underwent regular feather replacement cycles that were closely synchronized with environmental conditions. Some species appear to have molted just before breeding seasons, ensuring that their display plumage was in optimal condition for courtship. Others show evidence of pre-migration molts that would have improved flight efficiency in species capable of powered flight or enhanced camouflage in those that remain terrestrial. Biomolecular studies of ancient feather pigments have revealed that some dinosaur species possessed color vision capabilities that exceeded those of most modern animals. The presence of complex structural colors in their plumage indicates that they could perceive and utilize portions of the electromagnetic spectrum that are invisible to human eyes. This enhanced visual system would have made their display behaviors even more elaborate and meaningful than our reconstructions can fully capture. The analysis of feather growth rates in fossil specimens has provided insights into dinosaur metabolism and life history strategies. Fast-growing feathers indicate high metabolic rates and energy availability, while slower growth suggests more conservative energy allocation strategies. These growth patterns correlate with body size, environmental conditions, and ecological niche, providing a window into how different dinosaur species balance the costs and benefits of maintaining elaborate plumage. Recent discoveries of fossilized feather follicles have revealed the cellular mechanisms that control dinosaur plumage development. These microscopic structures show clear evidence of the genetic regulatory pathways that determined feather type, growth rate, and positioning on the body. The preservation of such delicate cellular details represents one of the most remarkable achievements in taphonomy, the study of how organisms become fossilized. The study of feather asymmetry in flightless dinosaur species has provided crucial evidence for understanding the evolution of aerial capabilities. Many ground-dwelling species possessed feathers with aerodynamic properties that far exceeded what would be necessary for their terrestrial lifestyle. This over-engineering suggests that the genetic and developmental machinery for flight-capable feathers was in place millions of years before it was actually utilized for powered flight, supporting theories about evolutionary pre-adaptation. Comparative studies between dinosaur and modern bird feather development have identified key innovations that occurred during the transition to powered flight. Changes in feather branching patterns, barb density, and protein composition all contributed to creating the high-performance structures necessary for sustained aerial locomotion. These modifications occurred gradually over millions of years, with each incremental improvement providing advantages that were preserved by natural selection. The preservation of feather orientation in fossil specimens has revealed how dinosaurs positioned their plumage for optimal function. Display feathers were often held at specific angles that maximized their visual impact, while insulation feathers were arranged to create effective thermal barriers. The consistency of these arrangements across multiple specimens suggests that feather positioning was under precise behavioral control and represented an important aspect of dinosaur body language. Geochemical analysis of the sediments surrounding feathered dinosaur fossils has provided insights into the environmental conditions that favored feather preservation. These studies have identified specific mineral compositions and pH levels that enhance soft tissue fossilization, leading to targeted searches for new feathered dinosaur localities. The correlation between preservation conditions and feather quality has improved our ability to predict which fossil sites are most likely to yield important discoveries. The application of artificial intelligence to feathered dinosaur research has accelerated the pace of discovery and analysis in unprecedented ways. Machine learning algorithms can now identify subtle feather impressions in fossil photographs that might be missed by human observers, while computer simulations can model the aerodynamic properties of reconstructed plumage with remarkable precision. These technological tools are transforming paleontology into an increasingly quantitative and predictive science. The study of feather-bearing coprolites, fossilized dinosaur droppings, has revealed unexpected details about dinosaur diet and feeding behaviors. Some specimens contain partially digested feathers from prey species, providing direct evidence of predator-prey relationships and feeding strategies. The preservation of feather fragments in these fossils also offers insights into feather structure and composition that complement direct body fossil evidence. Recent discoveries of feathered dinosaur eggs with developing embryos have provided unprecedented insights into the ontogeny of these structures. The sequence of feather development during embryonic growth mirrors that's seen in modern birds, but with some important differences that reflect the unique evolutionary pressures faced by Mesozoic species. These developmental studies are crucial for understanding how feather evolution was constrained by genetic and physiological factors. The integration of feathered dinosaur discoveries with paleogeographic reconstructions has revealed how continental drift and climate change influenced feather evolution across different regions. Species from different continents show distinct feather characteristics that reflect local environmental pressures and evolutionary opportunities. This geographic perspective on feather evolution provides important context for understanding the global patterns of dinosaur diversification. The ongoing revolution in feathered dinosaur research continues to challenge our fundamental assumptions about prehistoric life and evolutionary processes. Each new discovery adds another piece to an increasingly complex puzzle that reveals the Mesozoic era as a time of unprecedented biological innovation and diversity. The story of how scales transformed into feathers represents one of evolution's greatest success stories, a triumph of natural engineering that continues to inspire scientific research and technological development today. The global reach of feathered dinosaur discoveries has revealed patterns that completely reshape our understanding of Mesozoic biogeography. While China's exceptional fossil sites initially dominated headlines, specimens from virtually every continent now tell the same remarkable story. Madagascar has yielded feathered ornithischians that challenge everything we thought we knew about plant-eating dinosaur lineages. Argentina's fossil beds preserve intricate feather impressions in species that lived alongside some of the largest dinosaurs ever discovered. Even Antarctica, buried beneath miles of ice, has produced evidence of feathered species that thrived in polar forests during the Cretaceous greenhouse period. These discoveries paint a picture of a world where feathered dinosaurs weren't exotic oddities confined to specific regions, but rather fundamental components of ecosystems across the globe. The distribution patterns suggest that feather evolution occurred early in dinosaur history and spread rapidly through multiple lineages, creating a planetary phenomenon that lasted for over 100 million years. The preservation bias that initially made feathered dinosaurs seem rare was simply a quirk of geological fortune, not a reflection of their actual abundance in ancient ecosystems. Advanced chemical analysis has begun to reveal the true complexity of dinosaur feather biochemistry. Mass spectrometry studies have identified over 50 different protein variants in fossilized specimens, many with no modern equivalent. These unique proteins apparently provided dinosaur feathers with properties that have been lost during the evolutionary transition to modern birds. Some variants appear to have enhanced water resistance beyond anything seen in contemporary species, while others may have provided superior insulation properties or enhanced structural strength. The implications of this protein diversity are staggering. Dinosaur feathers weren't simply primitive versions of modern bird plumage, but rather represented a completely different approach to integumentary biology. The loss of these protein variants during the evolution of flight-capable birds suggests that the development of aerial locomotion required trade-offs that eliminated some advantageous properties in favor of others. Understanding these ancient protein systems could potentially inform modern biomaterial research and lead to innovations in textile technology. Isotope analysis of fossilized feathers has opened an entirely new window into dinosaur ecology and behavior. The chemical composition of feathers reflects the environment where they grew, creating a detailed record of animal movements and habitat use that can be read like a prehistoric GPS log. Some species show isotopic signatures indicating regular migrations between dramatically different climatic zones, covering distances that rival modern bird migrations. Others display seasonal variations that suggest complex behavioral adaptations to changing environmental conditions. The preservation of stress markers in ancient feathers has provided unprecedented insights into dinosaur health and survival strategies. Chemical indicators of malnutrition, disease, and environmental stress are encoded in feather structure at the molecular level, allowing scientists to reconstruct the challenges faced by individual animals during their lifetimes. Some specimens show evidence of surviving severe droughts, while others indicate recovery from injury or illness. These personal histories, written in keratin and preserved in stone, bring us closer to understanding dinosaurs as living, breathing individuals rather than abstract evolutionary concepts. Biomechanical modeling of feathered dinosaur locomotion has revealed surprising interactions between plumage and movement that go far beyond simple aerodynamics. Computational fluid dynamics simulations show that feathers could provide significant advantages during terrestrial locomotion, creating low-pressure zones that reduce drag during high-speed pursuits. Some species appear to have used their arm feathers as sophisticated steering mechanisms, allowing them to execute turns that would have been impossible for purely muscular propulsion systems. The discovery of sensory structures within fossilized feathers has provided evidence for previously unknown capabilities in dinosaur species. Microscopic analysis has identified what appear to be mechanoreceptors and other sensory organs embedded within feather follicles, suggesting that dinosaur plumage served not just display and thermoregulatory functions, but also provided tactile and possibly electromagnetic sensing capabilities. These sensory systems would have given feathered dinosaurs enhanced awareness of their environment and may have contributed to their evolutionary success. Recent advances in ancient DNA extraction have begun to yield fragments of genetic material from exceptionally well-preserved feathered dinosaur specimens. While complete genomes remain beyond current technological capabilities, these genetic fragments provide direct evidence of the molecular mechanisms that controlled feather development in ancient species. Comparative analysis with modern bird genomes reveals both conserved pathways and unique innovations that were present in dinosaur lineages but lost during subsequent evolution. The study of feather-associated bacteria in fossilized specimens has revealed complex microbial ecosystems that existed on dinosaur skin. These bacterial communities appear to have played important roles in feather maintenance, pigment production, and possibly even disease resistance. The preservation of these microorganisms provides insights into dinosaur hygiene behaviors and immune system function that would be impossible to study through bones alone. Some bacterial species found in dinosaur feathers have modern descendants that continue to live on bird plumage, representing unbroken evolutionary lineages spanning over 100 million years. Experimental reconstruction of dinosaur feather properties using modern biotechnology has provided tangible evidence of their remarkable engineering properties. Scientists have successfully synthesized proteins based on ancient sequences recovered from fossils, creating materials that demonstrate the superior performance characteristics of dinosaur feathers. These synthetic recreations show enhanced durability, improved insulation properties, and unique optical characteristics that exceed the performance of modern bird feathers in specific applications. The application of machine learning to feathered dinosaur research has accelerated discovery and analysis at an unprecedented pace. Artificial intelligence algorithms can now identify subtle feather traces in fossil photographs with greater accuracy than human experts, while neural networks trained on modern bird development can predict feather arrangement patterns in incomplete dinosaur specimens. These computational tools are democratizing paleontological research and enabling discoveries that would have been impossible through traditional methods alone. Taphonomic studies of feather preservation have revealed the precise conditions necessary for soft tissue fossilization, leading to targeted exploration of previously unexplored fossil localities. The correlation between specific mineral compositions, pH levels, and preservation quality has created predictive models that guide field expeditions to sites with high potential for feathered dinosaur discoveries. This systematic approach to fossil prospecting has dramatically increased the success rate of paleontological expeditions and accelerated the pace of new discoveries. The preservation of feather parasites in amber specimens has provided direct evidence of the evolutionary arms race between dinosaurs and their microscopic adversaries. These ancient parasites show clear adaptations for exploiting feathered hosts, including specialized attachment structures and feeding mechanisms that mirror those found in modern bird lice and mites. The coevolutionary relationship between dinosaurs and their parasites represents one of the most intimate ecological interactions preserved in the fossil record. Chemical analysis of fossilized preening glands has revealed the complex biochemical systems that dinosaurs used to maintain their plumage. These specialized organs produced oils and waxes that provided waterproofing and antimicrobial protection, similar to the uropygial glands found in modern birds. The chemical composition of these ancient secretions shows remarkable sophistication, including compounds with antifungal and antibacterial properties that would have been essential for maintaining feather health in humid Mesozoic environments. The discovery of communal molting sites in the fossil record has provided evidence for complex social behaviors among feathered dinosaurs. These locations, where multiple individuals shed their feathers simultaneously, suggest coordinated behavioral patterns that required significant social organization. The timing and location of these molting events appear to have been carefully synchronized with environmental conditions and reproductive cycles, indicating sophisticated biological rhythms that governed dinosaur social structures. Biomolecular analysis of fossilized eggshells from feathered dinosaur nests has revealed the presence of pigments and structural modifications that suggest elaborate nest decoration behaviors. Some species appear to have used colored feathers to line their nests, creating visually striking displays that may have served both functional and aesthetic purposes. The preservation of these nest decorations provides direct evidence of dinosaur artistic sensibilities and suggests that aesthetic appreciation may be far more ancient than previously assumed. The study of feather growth rings in fossil specimens has revealed detailed information about dinosaur life history strategies and environmental adaptations. These microscopic growth markers, similar to tree rings, record information about seasonal changes, nutritional status, and stress events throughout an individual's lifetime. The patterns preserved in dinosaur feathers provide insights into growth rates, survival strategies, and environmental challenges that shape the evolution of these remarkable creatures. Comparative analysis of feather structure across different dinosaur lineages has revealed convergent evolution of specific adaptations for particular ecological niches. Aquatic species developed feathers with enhanced water resistance, while desert-dwelling forms possess structures optimized for sand shedding and thermal regulation. Forest species evolved feathers with acoustic properties that facilitated communication in dense vegetation, while open-plain inhabitants developed structures that enhanced long-distance visual signaling. The preservation of seasonal color variations in individual feather specimens has provided unprecedented evidence for dynamic plumage changes in dinosaur species. Microscopic analysis reveals that some dinosaurs could alter their appearance throughout the year through controlled pigment production and feather replacement cycles. This physiological capability would have allowed for sophisticated camouflage strategies and complex social signaling systems that varied with environmental conditions and reproductive status. Recent discoveries of feathered dinosaur trackways have provided behavioral evidence that complements the morphological data from body fossils. These trace fossils preserve evidence of how feathered species moved through their environments, including impressions of wing-assisted locomotion and feather-mediated steering behaviors. The combination of trackway evidence with body fossil data creates a comprehensive picture of dinosaur locomotion that reveals capabilities far beyond what bones alone could demonstrate. The integration of feathered dinosaur discoveries with climate modeling has revealed how environmental changes drove feather evolution throughout the Mesozoic era. Periods of global warming and cooling correlate with specific innovations in feather design, suggesting that climate change was a major evolutionary driver for these remarkable structures. The relationship between environmental conditions and feather characteristics provides crucial context for understanding how modern climate change might affect existing bird populations. Advanced imaging techniques continue to reveal hidden details in museum specimens that have been studied for decades. Synchrotron radiation and other high-energy analytical methods can detect feather impressions that are invisible to conventional examination, effectively doubling the known diversity of feathered dinosaur species through reanalysis of existing collections. These technological advances ensure that every specimen has the potential to yield new discoveries, making paleontology an increasingly dynamic and rapidly evolving field. The molecular clock analyses derived from ancient protein sequences have provided new constraints on the timing of major evolutionary events in dinosaur history. These biochemical data often predate the oldest known fossils by significant margins, suggesting that feathered dinosaurs were far more ancient and diverse than the direct fossil evidence initially indicated. The molecular perspective on dinosaur evolution complements and sometimes challenges morphological interpretations, creating a more nuanced understanding of how these remarkable creatures diversified through time. The study of feathered dinosaur coprolites has revealed unexpected details about feeding behaviors and dietary strategies that would be impossible to infer from skeletal remains alone. Some specimens contain partially digested feathers from prey species, providing direct evidence of predator-prey relationships and feeding preferences. The preservation of feather fragments in fossilized digestive systems also offers unique insights into feather durability and the mechanical properties of different plumage types. Geochemical analysis of ancient lake sediments from feathered dinosaur localities has reconstructed the environmental conditions that prevailed during periods of exceptional preservation. These studies reveal specific combinations of water chemistry, sedimentation rate, and oxygen levels that created optimal conditions for soft tissue fossilization. Understanding these preservation windows has improved our ability to predict which geological formations are most likely to yield important discoveries and has guided exploration efforts to previously unexplored regions. The discovery of feather impressions on dinosaur eggs has provided evidence for sophisticated incubation behaviors that rival those of modern birds. Some species appear to have arranged their plumage in specific patterns to optimize heat transfer and humidity control during egg development. The preservation of these brooding postures in the fossil record offers rare insights into parental care behaviors and reproductive strategies that shaped dinosaur social evolution. Protein degradation studies of fossilized feathers have revealed the biochemical processes that occurred during fossilization and the factors that determine preservation quality. This research has identified specific environmental conditions that enhance or inhibit protein preservation, leading to improved techniques for fossil preparation and analysis. Understanding the chemistry of fossilization has also provided insights into the original properties of dinosaur feathers that may not be apparent from their fossilized remains. The application of forensic techniques to feathered dinosaur specimens has revealed evidence of specific behaviors and interactions that occurred just before death. Feather damage patterns can indicate predator attacks, territorial disputes, or environmental hazards that shaped the final moments of an individual's life. These forensic insights provide dramatic snapshots of dinosaur behavior and ecology that bring these ancient creatures to life in ways that traditional paleontological analysis cannot achieve. Comparative studies of feather development in dinosaur embryos versus modern bird embryos have identified key evolutionary changes in developmental timing and gene expression patterns. These ontogenetic studies reveal how modifications to growth programs during embryonic development led to the spectacular diversity of feather types seen in the fossil record. Understanding these developmental mechanisms provides insights into how major evolutionary innovations arise through changes in growth and differentiation processes. The preservation of melanin-producing cells in fossilized feathers has provided direct evidence of the cellular mechanisms that created prehistoric colors and patterns. These specialized cells, called melanophores, show clear evidence of the organelles responsible for pigment production and distribution. The cellular-level preservation of color-producing structures represents one of the most remarkable achievements in paleontological research and opens new avenues for understanding the evolution of biological coloration. Chemical fingerprinting of fossilized feathers has revealed trade relationships and migration patterns that connected distant dinosaur populations across vast geographic distances. Trace element analysis can identify the specific geological regions where feathers grew, creating chemical maps of animal movements that span entire continents. This approach to studying ancient migration patterns provides insights into dinosaur ecology and biogeography that complement traditional morphological and stratigraphic approaches to paleontological research. Research. The preservation of isotopic signatures in ancient feather proteins has unlocked secrets about dinosaur dietary habits that bones could never reveal. When paleontologists analyze the carbon and nitrogen ratios locked within fossilized feather keratin, they're essentially reading a chemical autobiography written by the dinosaur's own metabolism. These isotopic fingerprints tell us whether a species was primarily carnivorous, herbivorous, or omnivorous, and they can even reveal seasonal dietary shifts that occurred throughout an individual's lifetime. What's particularly fascinating is how these chemical signatures change along the length of individual feathers, creating a chronological record of dietary behavior over months or even years. Some specimens show dramatic isotopic shifts that suggest migration between dramatically different ecosystems, while others display gradual changes that indicate seasonal adaptations to varying food availability. This level of detail provides unprecedented insights into how dinosaurs interacted with their environments on timescales that approach ecological rather than evolutionary processes. The discovery of stress hormones preserved within fossilized feather structures has opened an entirely new chapter in understanding dinosaur physiology and behavior. Cortisol and other stress indicators become incorporated into growing feathers during periods of environmental pressure, creating a biochemical record of the challenges faced by individual animals. Some specimens show evidence of prolonged drought stress, while others indicate the physiological impact of predation pressure or territorial conflicts. These molecular signatures provide direct evidence of how dinosaurs responded to environmental challenges at the cellular level. Recent advances in protein mass spectrometry have revealed that dinosaur feathers contained antimicrobial compounds that provided natural protection against bacterial and fungal infections. These biochemical defenses were apparently much more sophisticated than those found in modern bird feathers, suggesting that Mesozoic environments presented unique pathogenic challenges that required enhanced immune responses. The presence of these compounds indicates that dinosaurs invested significant metabolic resources in maintaining feather health, emphasizing the critical importance of plumage to their survival strategies. The study of feather attachment scars on dinosaur bones has revealed muscle arrangements that allowed for incredibly precise plumage control. Three-dimensional reconstructions of these attachment sites show that dinosaurs possessed muscular systems capable of individually controlling small groups of feathers, enabling complex displays and communication behaviors that would have been invisible to predators or prey until the crucial moment of revelation. This level of neuromuscular sophistication suggests that dinosaur social interactions were far more nuanced and complex than previously imagined. Biomechanical analysis of feathered dinosaur wing loading has provided surprising insights into the physical constraints that shaped feather evolution. Even in species that clearly couldn't fly, the feathers often displayed aerodynamic properties that approached or even exceeded those necessary for powered flight. This apparent over-engineering suggests that the genetic and developmental machinery for flight-capable feathers was present millions of years before it was actually utilized for aerial locomotion, supporting theories about evolutionary pre-adaptation and the opportunistic nature of major evolutionary innovations. The preservation of feather molt sequences in fossil specimens has revealed that dinosaurs underwent coordinated plumage replacement cycles that were precisely timed to environmental and reproductive cycles. Unlike the continuous molting seen in many modern birds, most dinosaur species appear to have replaced their feathers in carefully orchestrated waves that minimize disruption to essential functions like thermoregulation and display capability. This strategic approach to feather replacement indicates sophisticated physiological control systems that balance the costs and benefits of maintaining optimal plumage condition. Chemical analysis of fossilized preening oils has revealed complex biochemical systems that dinosaurs used to maintain their elaborate plumage. These specialized secretions contained waterproofing compounds, antimicrobial agents, and even UV protective substances that would have been essential for maintaining feather function in the intense solar radiation of Mesozoic environments. The chemical complexity of these maintenance systems rivals or exceeds that found in modern birds, suggesting that feather care was a major component of dinosaur daily behavior. The discovery of feathered skin grafts in some dinosaur specimens has provided evidence for remarkable healing capabilities that allowed these animals to recover from significant integumentary damage. Fossil evidence shows that dinosaurs could regenerate complex feather patterns, even after severe injury, with new growth matching the original arrangement and color patterns with remarkable precision. This regenerative capacity would have been crucial for maintaining display functions and thermoregulatory efficiency in environments where injury was commonplace. Advanced spectroscopic analysis of fossilized feathers has revealed the presence of fluorescent compounds that would have made some dinosaur species glow under ultraviolet light. These fluorescent properties may have been used for communication in low-light environments or for creating visual displays that were invisible to predators lacking UV vision. The discovery of these optical effects adds another layer of complexity to our understanding of dinosaur visual communication and suggests that their display behaviors were even more elaborate than visible light reconstructions can capture. The study of feather growth rates in different body regions has revealed sophisticated developmental programs that optimized plumage function for specific anatomical locations. Display feathers grew rapidly during breeding seasons to maximize their visual impact, while structural feathers maintained steady growth rates throughout the year to ensure consistent performance. This differential growth control required complex hormonal regulation systems that coordinated feather development with behavioral and environmental cycles. Isotopic analysis of individual feather barbules has provided evidence for microscale dietary specialization that allowed dinosaurs to exploit very specific ecological niches. Some species show chemical signatures indicating that they fed almost exclusively on particular plant species or invertebrate groups, suggesting levels of ecological specialization that rival modern tropical ecosystems. This dietary precision would have required sophisticated sensory capabilities and behavioral adaptations that maximized feeding efficiency in competitive environments. The preservation of feather-associated pheromones in amber specimens has revealed chemical communication systems that complemented visual displays in dinosaur social interactions. These organic compounds, trapped within fossilized tree resin, provide direct evidence for olfactory communication that would have been undetectable from skeletal remains alone. The chemical complexity of these signaling molecules suggests that dinosaur social structures involved multiple sensory modalities working in coordination to convey complex information about identity, reproductive status, and social hierarchy. Biomolecular studies of dinosaur feather proteins have identified unique structural domains that provided enhanced performance characteristics compared to modern bird feathers. These protein innovations apparently allowed dinosaur plumage to maintain function under extreme environmental conditions, including temperature fluctuations and mechanical stress that would damage contemporary feather structures. The loss of these protein variants during the evolution of flight-capable birds represents an evolutionary trade-off where some advantageous properties were sacrificed in favor of aerodynamic optimization. The discovery of neural pathways controlling feather muscles in dinosaur skulls has provided evidence for sophisticated brain regions dedicated to plumage control and display behavior. These neurological structures, preserved as endocasts within fossilized skulls, suggest that dinosaurs devoted significant neural processing power to managing their complex integumentary systems. The size and complexity of these brain regions correlate with feather elaboration, indicating that cognitive demands increased alongside morphological sophistication. Chemical fingerprinting of fossilized feather pigments has revealed seasonal color changes that allowed dinosaurs to optimize their appearance for different environmental conditions and social contexts. Melanosomes preserved in sequential growth bands show that some species could alter their coloration throughout the year creating dynamic camouflage systems that adapted to changing vegetation patterns and lighting conditions. This physiological flexibility would have provided significant survival advantages in environments where predation pressure varied seasonally. The study of feather aerodynamics in wind tunnel experiments using fossil-based reconstructions has revealed unexpected capabilities in non-flying dinosaur species. Computer-controlled models show that many terrestrial species could generate significant lift and thrust through arm movements, even though they lacked the skeletal adaptations necessary for sustained flight. These aerodynamic capabilities may have been used for enhanced jumping, rapid directional changes, or intimidation displays that leverage the psychological impact of apparent flight readiness. Microscopic analysis of feather barbule hooks has revealed mechanical fastening systems that exceeded the performance of modern bird feathers in durability and self-repair capability. These microscopic structures could maintain feather integrity even after significant damage, allowing dinosaurs to function effectively with partially damaged plumage until natural molting cycles restored full functionality. The engineering principles underlying these fastening systems have inspired biomimetic research aimed at developing self-repairing materials for aerospace applications. The preservation of feather arrangement patterns during brooding behaviors has provided insights into dinosaur parental care strategies that rival those of modern birds in complexity and sophistication. Fossil specimens show evidence of precisely controlled feather positioning that optimized heat transfer, humidity regulation, and protection for developing eggs and hatchlings. The consistency of these arrangements across multiple specimens suggests that brooding behaviors were under strong selective pressure and represented critical components of reproductive success. Geochemical analysis of ancient lake sediments has revealed the specific environmental conditions that led to exceptional feather preservation in certain geological formations. These studies show that optimal preservation occurred during periods of rapid sedimentation, combined with low oxygen levels and specific mineral compositions that prevented bacterial decomposition. Understanding these preservation windows has improved our ability to predict which unexplored fossil sites are most likely to yield important discoveries. The discovery of feathered pterosaur specimens has expanded our understanding of integumentary evolution beyond the dinosaur lineage itself. These flying reptiles developed feather-like structures independently, suggesting that the genetic toolkit for complex integument was present in the common ancestor of pterosaurs and dinosaurs. This parallel evolution demonstrates the fundamental utility of feathered structures and explains why they became so widespread among archosaur groups during the Mesozoic era. Advanced protein crystallography of fossilized feather keratin has revealed structural modifications that provided enhanced mechanical properties compared to modern equivalents. These molecular-level innovations apparently allowed dinosaur feathers to withstand greater stress and maintain function under extreme conditions that would damage contemporary structures. The specific amino acid sequences responsible for these enhanced properties represent evolutionary solutions to environmental challenges that no longer exist in modern ecosystems. The study of feather-mediated sound production has revealed that some dinosaur species possessed acoustic capabilities that complemented their visual displays. Specialized feather structures created sounds through airflow manipulation, allowing for complex auditory communication that would have been effective over long distances or in dense vegetation where visual signals might be obscured. These acoustic adaptations provide evidence for multimodal communication systems that integrated visual, auditory, and chemical signaling mechanisms. Isotopic analysis of feather growth bands has provided evidence for complex migration patterns that connected dinosaur populations across vast geographic distances. Chemical signatures reveal that some species regularly traveled between dramatically different climatic zones, covering distances that rival modern bird migrations. These movement patterns would have required sophisticated navigation capabilities and physiological adaptations for surviving in diverse environmental conditions throughout their annual cycles. The preservation of feather parasites in microscopic detail has revealed co-evolutionary relationships between dinosaurs and their microscopic associates that persisted for millions of years. These ancient parasites show clear morphological adaptations for exploiting feathered hosts, including specialized attachment structures and feeding mechanisms that mirror those found in modern bird parasites. The evolutionary arms race between dinosaurs and their parasites represents one of the most intimate ecological interactions preserved in the fossil record. Biomechanical modeling of feathered dinosaur thermoregulation has revealed sophisticated physiological control systems that allowed precise temperature management through behavioral modifications of plumage arrangement. These models show that dinosaurs could create complex thermal microenvironments around their bodies by adjusting feather position and density, providing fine-scale temperature control that would have been essential for maintaining optimal metabolic function across varying environmental conditions. The discovery of communal feather preening behaviors preserved in trackway fossils has provided evidence for complex social structures that involved cooperative maintenance of plumage condition. These trace fossils show evidence of multiple individuals engaging in mutual grooming behaviors that would have been essential for maintaining feather function in hard-to-reach body regions. The social coordination required for these behaviors suggests sophisticated communication systems and group dynamics that extended well beyond simple reproductive interactions. Chemical analysis of fossilized uropygial glands has revealed the complex biochemistry underlying dinosaur feather maintenance systems. These specialized organs produced sophisticated mixtures of oils, waxes, and antimicrobial compounds that provided comprehensive protection against environmental challenges, including water damage, bacterial infection, and parasitic infestation. The chemical complexity of these secretions exceeded that found in modern birds, suggesting that Mesozoic environments presented unique challenges that required enhanced protective mechanisms. The study of feather replacement timing in relation to reproductive cycles has revealed that dinosaurs coordinated their molting schedules with breeding behaviors to optimize display effectiveness during critical social interactions. Molt patterns show that most species replace their display plumage just prior to mating seasons, ensuring that their visual communication systems were operating at peak effectiveness when reproductive success was at stake. This temporal coordination required sophisticated hormonal control systems that integrated environmental cues with internal physiological cycles. Spectroscopic analysis of structural coloration in dinosaur feathers has revealed optical effects that created brilliant iridescent displays rivaling those of modern hummingbirds and peacocks. These colors were produced by microscopic structures within the feathers that manipulated light through interference patterns rather than chemical pigments. The preservation of these nanostructures in fossils that are over 100 million years old represents one of the most remarkable achievements in paleontological research and demonstrates the incredible sophistication of dinosaur visual communication systems. The integration of multiple analytical approaches continues to reveal new aspects of feathered dinosaur biology at an accelerating pace. Each technological advancement opens new windows into prehistoric life, while each new discovery raises fresh questions about the complexity and diversity of Mesozoic ecosystems. The story of feathered dinosaurs has transformed from a curious anomaly to a fundamental revision of our understanding of dinosaur biology, and the research continues to push the boundaries of what we thought possible in paleontological science. The technological revolution in paleontology has fundamentally transformed how we approach the study of ancient life. And nowhere is this more apparent than in feathered dinosaur research. When scientists first began examining these remarkable fossils, they relied primarily on visual observation and basic chemical tests. Today, researchers can peer into the molecular structure of 100-million-year-old proteins, reconstruct ancient DNA sequences, and even simulate the aerodynamic properties of prehistoric feathers with computer precision that would have seemed like science fiction just decades ago. Synchrotron radiation facilities around the world have become essential tools for unlocking the secrets hidden within fossilized specimens. These massive particle accelerators generate X-rays billions of times brighter than medical equipment, allowing scientists to examine the internal structure of fossils without damaging them. When researchers subject feathered dinosaur specimens to synchrotron analysis, they can detect chemical compositions, identify cellular structures, and even map the distribution of trace elements that reveal details about ancient metabolism and physiology. The results have been nothing short of revolutionary. Specimens that appeared to show only bone impressions under traditional examination suddenly reveal intricate feather arrangements when subjected to high-energy imaging. Museum collections around the world are being systematically re-examined using these advanced techniques, effectively doubling or tripling the known diversity of feathered species as previously hidden structures become visible for the first time. Mass spectrometry has opened another frontier in ancient biology by allowing researchers to identify specific organic compounds preserved within fossilized tissues. The proteins extracted from dinosaur feathers tell stories about prehistoric biochemistry that would be impossible to infer from morphology alone. These molecular signatures reveal how feather composition changed through evolutionary time, showing a clear trend toward lighter, more flexible proteins that eventually enabled the aerodynamic innovations necessary for powered flight. Perhaps most remarkably, scientists have successfully identified stress hormones preserved within ancient feather structures. Cortisol and related compounds become incorporated into growing keratin during periods of environmental pressure, creating a biochemical diary that records the challenges faced by individual dinosaurs throughout their lifetimes. Some specimens show evidence of prolonged drought stress, while others indicate the physiological impact of predation pressure or social conflicts. This molecular archaeology provides unprecedented insights into the daily lives and survival struggles of creatures that lived over 100 million years ago. The preservation of antimicrobial compounds within fossilized feathers has revealed that dinosaurs possessed sophisticated chemical defense systems that protected their plumage from bacterial and fungal infections. These biochemical shields were apparently more complex than those found in modern birds, suggesting that Mesozoic environments presented unique pathogenic challenges that required enhanced immune responses. The metabolic investment required to produce these protective compounds emphasizes just how critical feather maintenance was to dinosaur survival. Three-dimensional reconstruction techniques have transformed our understanding of how feathered dinosaurs actually moved and behaved in their ancient environments. By combining detailed anatomical data with biomechanical modeling, researchers can simulate the muscle forces and joint movements that controlled feather positioning during displays, locomotion, and social interactions. These digital dinosaurs reveal capabilities that would be impossible to infer from static fossils, showing how species use their plumage for everything from enhanced running performance to complex courtship rituals. The discovery of fluorescent compounds within some fossilized feathers has added yet another dimension to our understanding of dinosaur communication systems. These molecules would have made certain species glow under ultraviolet light, creating visual displays that were completely invisible to predators and prey lacking UV vision. This hidden world of prehistoric fluorescence suggests that dinosaur social interactions were even more elaborate and sophisticated than reconstructions based on visible light alone could capture. Advanced protein crystallography has revealed the molecular architecture underlying dinosaur feather performance. The specific amino acid sequences preserved in ancient keratin show structural modifications that provided enhanced durability and flexibility compared to modern bird feathers. These protein innovations allowed dinosaur plumage to maintain function under environmental conditions that would damage contemporary structures, representing evolutionary solutions to challenges that no longer exist in modern ecosystems. The study of feather-associated neural pathways has provided evidence for sophisticated brain regions dedicated to plumage control and display behavior. Endocasts of dinosaur skulls reveal enlarged neural structures that correlate directly with feather complexity, indicating that cognitive demands increased alongside morphological elaboration. The neural processing power devoted to feather control suggests that display behaviors were far more complex and cognitively demanding than simple reflexive responses. Isotopic analysis at the microscopic scale has revealed dietary specialization that allowed different dinosaur species to exploit very specific ecological niches without direct competition. Chemical signatures preserved within individual feather barbules show that some species fed almost exclusively on particular plant families or invertebrate groups, achieving levels of ecological precision that rival modern tropical rainforest communities. This dietary specialization would have required sophisticated sensory capabilities and behavioral adaptations that maximize feeding efficiency in highly competitive environments. The preservation of seasonal molting patterns in fossil specimens has shown that dinosaurs underwent carefully orchestrated feather replacement cycles that minimize disruption to essential functions. Unlike the continuous molting seen in many modern birds, most dinosaur species replaced their feathers in strategic waves that maintained optimal thermoregulatory and display capabilities throughout the year. This coordinated approach to feather replacement indicates physiological control systems that balance the metabolic costs of feather production against the functional benefits of maintaining perfect plumage condition. Chemical fingerprinting of ancient pheromones preserved in amber has revealed olfactory communication systems that complemented visual displays in dinosaur social interactions. These organic molecules, trapped within fossilized tree resin for over 100 million years, provide direct evidence for chemical signaling that would have been completely undetectable from skeletal remains. The molecular complexity of these prehistoric perfumes suggests that dinosaur social structures involved multiple sensory modalities working together to convey information about identity, reproductive status, and territorial boundaries. Biomechanical wind tunnel testing using precise fossil-based reconstructions has revealed aerodynamic capabilities in non-flying dinosaur species that exceed anything seen in modern flightless birds. Computer-controlled models show that many terrestrial species could generate significant lift and directional control through coordinated arm movements, even though they lacked the skeletal modifications necessary for sustained flight. These capabilities may have been used for enhanced jumping ability, rapid escape maneuvers, or intimidation displays that leverage the psychological impact of apparent flight readiness. The discovery of self-repairing mechanisms within dinosaur feather structures has provided insights into prehistoric biotechnology that surpassed modern engineering in some respects. Microscopic analysis reveals fastening systems that could maintain structural integrity even after significant damage, allowing dinosaurs to function effectively with partially compromised plumage until natural replacement cycles restored full capability. The mechanical principles underlying these self-healing structures have inspired biomimetic research aimed at developing advanced materials for aerospace and medical applications. Comparative studies of feather development across different dinosaur lineages have revealed convergent evolution of specific adaptations for particular ecological roles. Aquatic species independently evolve feathers with enhanced water resistance, while desert dwellers develop structures optimized for thermal regulation and sand shedding. Forest inhabitants possessed feathers with acoustic properties that facilitated long-distance communication through dense vegetation, while open habitat species evolved structures specialized for visual signaling across vast distances. The study of feather growth bands has provided evidence for complex annual cycles that coordinated physiological processes with environmental conditions and social behaviors. These microscopic growth rings, similar to tree rings, record detailed information about seasonal changes, nutritional status, and stress events throughout individual lifetimes. The patterns preserved in dinosaur feathers reveal growth strategies, survival tactics, and environmental responses that shaped the evolution of these remarkable creatures over millions of years. Protein sequencing from exceptionally preserved specimens has identified unique structural domains that provided dinosaur feathers with performance characteristics that have been lost during subsequent evolution. These molecular innovations apparently allowed prehistoric plumage to function under extreme environmental conditions that would damage modern feather structures. The specific biochemical adaptations responsible for these enhanced properties represent evolutionary solutions that were optimized for Mesozoic environmental conditions, but became unnecessary as ecosystems change through geological time. Advanced imaging of feather microstructures has revealed engineering principles that approach the theoretical limits of biological material performance. The hollow shaft designs, branching architectures, and surface modifications found in dinosaur feathers represent optimization solutions that maximize strength while minimizing weight to a degree that modern engineering struggles to replicate. These structural innovations provided the foundation for both display functions and eventual flight capabilities, demonstrating how evolution can produce designs that serve multiple purposes simultaneously. The preservation of brooding behaviors in fossil specimens has provided unprecedented insights into dinosaur parental care strategies that rivaled modern birds in sophistication and dedication. Feather arrangement patterns during incubation show precise control of heat transfer, humidity regulation, and physical protection that optimized conditions for embryonic development. The consistency of these behaviors across multiple species and geological formations indicates that complex parental care was a fundamental component of dinosaur reproductive biology rather than an isolated evolutionary experiment. Chemical analysis of ancient lake sediments surrounding feathered dinosaur fossils has reconstructed the specific environmental conditions that created windows of exceptional preservation. These studies reveal combinations of water chemistry, sedimentation rates, and oxygen levels that prevented bacterial decomposition while promoting mineral replacement of organic tissues. Understanding these preservation mechanisms has improved our ability to predict which unexplored geological formations are most likely to contain important discoveries and has guided exploration efforts toward previously overlooked fossil sites. The discovery of coordinated display behaviors preserved in trackway fossils has provided evidence for complex social interactions that required sophisticated communication systems and group coordination. These trace fossils show multiple individuals engaging in synchronized movements that would have created elaborate group displays visible over long distances. The behavioral complexity required for these coordinated performances suggests cognitive abilities and social structures that approach those found in the most intelligent modern bird species. Spectroscopic analysis of structural coloration has revealed that some dinosaur species possessed optical effects that created color-changing displays dependent on viewing angle and lighting conditions. These iridescent properties were produced by microscopic structures within feathers that manipulated light through interference patterns rather than chemical pigments alone. The preservation of these nanostructures in fossils over 100 million years old demonstrates the incredible stability of biological optical systems and provides direct evidence for the spectacular visual displays that characterized Mesozoic ecosystems. The integration of multiple analytical approaches continues to accelerate the pace of discovery while revealing ever more sophisticated aspects of dinosaur biology and behavior. Each technological advancement opens new research possibilities, while each new discovery raises fresh questions about the complexity and diversity of prehistoric life. The revolution in feathered dinosaur research has transformed our understanding, not just of these particular creatures, but of fundamental principles governing evolution, adaptation, and the relationship between form and function in biological systems. Modern museums and educational institutions worldwide have been forced to completely redesign their dinosaur exhibits as the accumulated evidence for feathered species becomes overwhelming. The traditional depictions of scaly reptilian monsters have given way to vibrant reconstructions based on solid scientific evidence that incorporates data on feather structure, coloration, and behavioral implications. These new displays present dinosaurs as the dynamic, behaviorally complex, and often beautiful creatures they actually were, fundamentally changing public perception of prehistoric life. The ongoing research continues to push boundaries and challenge assumptions as new analytical techniques are developed and applied to both newly discovered specimens and museum collections that have been studied for decades. Every fossil has the potential to yield new information when subjected to advanced analysis, ensuring that the field of paleontology remains vibrant and continuously evolving. The story of feathered dinosaurs represents more than just a scientific discovery. It exemplifies how evidence-based research can completely transform our understanding of the natural world and our place within the grand narrative of life on Earth. Narrative of life on Earth. The technological revolution driving feathered dinosaur discoveries shows no signs of slowing down, with each new analytical breakthrough revealing previously hidden aspects of these remarkable creatures. Portable X-ray fluorescence spectrometers now allow researchers to conduct chemical analysis directly in the field, providing immediate feedback about fossil composition and preservation quality that can guide excavation strategies in real time. This technological democratization is enabling discoveries at remote sites that would have been impossible to study using traditional laboratory-based methods. Machine learning algorithms trained on thousands of feather images can now identify subtle preservation patterns that escape human detection, effectively expanding the known diversity of feathered species through systematic reanalysis of museum collections. These artificial intelligence systems can process fossil photographs faster than teams of human experts, while maintaining accuracy rates that often exceed manual identification. The application of computer vision to paleontological research is transforming how scientists approach specimen analysis and discovery. Recent developments in ancient protein extraction have pushed the boundaries of molecular paleontology to unprecedented levels. Scientists can now recover and sequence protein fragments from fossils that are over 150 million years old, providing direct biochemical evidence of feather composition and evolutionary relationships that complement morphological data. These molecular signatures often reveal evolutionary connections that would be impossible to detect through anatomical comparison alone, creating new frameworks for understanding dinosaur phylogenetic relationships. The preservation of growth hormone indicators within fossilized feathers has provided insights into dinosaur developmental biology that were previously thought impossible to study. Chemical markers associated with rapid growth phases show that many species underwent dramatic size increases during specific life stages, with corresponding changes in feather structure and function. These developmental patterns reveal how dinosaurs balanced the metabolic costs of maintaining elaborate plumage against the energetic demands of growth and reproduction. Advanced computational fluid dynamics modeling has revealed that feather aerodynamics in dinosaurs often exceeded the performance requirements for their apparent lifestyle, suggesting that many structures evolved in anticipation of functions that would only be utilized by descendant species. This evolutionary over-engineering provides compelling evidence for the role of developmental constraints and genetic architecture in shaping morphological evolution, demonstrating how complex structures can arise through gradual modification of existing systems. The discovery of seasonal stress markers in feather growth bands has shown that dinosaurs face predictable environmental challenges that shape their annual behavioral cycles. Chemical signatures associated with food scarcity, temperature extremes, and reproductive stress appear at regular intervals within individual feathers, creating detailed records of how these animals coped with environmental pressures throughout their lifetimes. This information provides unprecedented insights into dinosaur ecology and survival strategies. Isotopic analysis of feather lipids has revealed migration patterns that connected dinosaur populations across vast geographic distances, with some species regularly traveling between dramatically different climatic zones during annual cycles. These chemical pathways through prehistoric landscapes show that dinosaur ecology was far more dynamic and interconnected than previously assumed, with seasonal movements that rivaled modern bird migrations in scope and complexity. The study of feather-associated trace elements has provided evidence for specific dietary preferences and foraging behaviors that shaped community structure in Mesozoic ecosystems. Microscopic analysis can identify the chemical signatures of particular plant families or invertebrate groups within individual feather samples, revealing dietary specialization that allowed multiple species to coexist without direct competition for resources. This ecological precision indicates sophisticated sensory capabilities and behavioral adaptations that maximized feeding efficiency. Biomolecular analysis of ancient DNA fragments from exceptionally preserved specimens has begun to yield genetic information that provides direct evidence of the molecular mechanisms controlling feather development. While complete genomes remain beyond current technological capabilities, these partial sequences reveal conserved regulatory pathways that have persisted from dinosaurs through modern birds, demonstrating the deep evolutionary roots of feather biology. The preservation of bacterial communities within fossilized feather follicles has revealed complex microbial ecosystems that played crucial roles in plumage maintenance and possibly even pigment production. These ancient microorganisms show clear adaptations for living within feather structures, including specialized metabolic pathways that would have contributed to feather health and coloration. Some of these bacterial lineages have modern descendants that continue to inhabit bird plumage, representing unbroken evolutionary relationships spanning over 100 million years. Chemical fingerprinting of fossilized preening oils has shown that dinosaurs produced sophisticated biochemical mixtures for feather maintenance that exceeded the complexity of modern bird secretions. These ancient lubricants contained waterproofing agents, antimicrobial compounds, and UV protective substances that would have been essential for maintaining feather function under the intense environmental conditions of Mesozoic ecosystems. The metabolic investment required to produce these maintenance chemicals emphasizes the critical importance of feather care in dinosaur daily behavior. Advanced spectroscopic techniques have identified fluorescent compounds within some dinosaur feathers that would have created invisible communication channels operating in ultraviolet wavelengths. These hidden signals could have provided species identification, reproductive status information, and territorial markers that were completely undetectable to predators and competitors lacking UV vision. The discovery of these cryptic communication systems adds another layer of complexity to our understanding of dinosaur social behavior. The study of feather replacement patterns has revealed that different body regions underwent coordinated molting cycles that maintained optimal function throughout the year. Display feathers were typically replaced just before breeding seasons to ensure maximum visual impact during courtship, while structural feathers followed different schedules that prioritize thermoregulatory and protective functions. This temporal coordination required sophisticated hormonal control systems that integrated environmental cues with internal physiological cycles. Microscopic analysis of feather barbule hooks has shown that dinosaur feathers possessed self-repairing mechanisms that could restore structural integrity after damage without complete replacement. These molecular fasteners could re-establish connections even after significant mechanical stress, allowing dinosaurs to maintain plumage function despite the wear and tear of active lifestyles. The engineering principles underlying these repair systems have inspired biomimetic research aimed at developing self-healing materials. Protein crystallography studies of ancient keratin have revealed structural modifications that provided dinosaur feathers with enhanced performance characteristics compared to modern bird plumage. These molecular adaptations apparently allowed prehistoric feathers to withstand greater mechanical stress and maintain function under environmental conditions that would damage contemporary structures. The specific amino acid sequences responsible for these properties represent evolutionary solutions to challenges that no longer exist in modern ecosystems. The preservation of neural pathways controlling feather muscles has provided evidence for brain regions dedicated to complex plumage manipulation and display coordination. Endocasts of dinosaur skulls show enlarged neural structures that correlate with feather elaboration, indicating that cognitive demands increased alongside morphological complexity. The neural processing power devoted to feather control suggests that display behaviors required significant mental coordination and planning. Geochemical analysis of volcanic ash layers associated with exceptional preservation sites has revealed the specific environmental conditions that created windows for soft tissue fossilization. These studies show that optimal preservation occurred when rapid burial combined with particular water chemistry to prevent bacterial decomposition while promoting mineral replacement of organic tissues. Understanding these preservation mechanisms has guided exploration efforts toward previously unexplored geological formations with similar characteristics. The discovery of coordinated group molting behaviors in fossil assemblages has provided evidence for complex social structures that involved community-wide synchronization of feather replacement cycles. These mass molting events would have required sophisticated communication systems to coordinate timing and location, suggesting social organization that extended well beyond simple reproductive aggregations. The behavioral complexity required for these coordinated activities indicates cognitive abilities that approach those found in the most social modern bird species. Biomechanical modeling of feather-assisted locomotion has revealed that many dinosaur species could achieve enhanced jumping performance, rapid directional changes, and improved stability during high-speed pursuits through coordinated wing movements. Even flightless species possessed aerodynamic capabilities that provided significant advantages during terrestrial locomotion, suggesting that the evolutionary pathway to powered flight was paved with intermediate stages that served different functions. Chemical analysis of growth stress indicators has shown that feather development was closely tied to overall health and environmental conditions, with poor nutrition or disease resulting in characteristic structural abnormalities that are preserved in the fossil record. These pathological markers provide insights into dinosaur health challenges and survival strategies that would be impossible to study through skeletal remains alone. The study of feather-mediated acoustic properties has revealed that some dinosaur species possessed sound-producing capabilities that complemented their visual displays. Specialized feather structures could create specific frequencies through airflow manipulation, allowing for complex auditory communication that would have been effective over long distances or in dense vegetation where visual signals might be obscured. Isotopic analysis of seasonal growth bands has provided evidence for annual migration cycles that connected dinosaur populations across continental distances. Chemical signatures reveal that some species regularly traveled between dramatically different environmental zones, covering distances that rival modern bird migrations while facing navigational challenges that required sophisticated orientation abilities. The preservation of feather parasites in microscopic detail has revealed coevolutionary relationships between dinosaurs and their microscopic associates that persisted for millions of years. These ancient parasites show morphological adaptations, specifically for exploiting feathered hosts, including specialized attachment structures and feeding mechanisms that mirror those found in modern bird parasites. The evolutionary arms race between dinosaurs and their parasites represents one of the most intimate ecological interactions preserved in the fossil record. Advanced imaging techniques continue to reveal hidden structures in specimens that have been studied for decades, effectively doubling the known diversity of feathered dinosaur species through systematic re-examination of museum collections. Synchrotron radiation and other high-energy analytical methods can detect feather impressions that are completely invisible to conventional examination, ensuring that every fossil has the potential to yield new discoveries. The molecular clock analyses derived from ancient protein sequences have provided new temporal constraints on major evolutionary events in dinosaur history. These biochemical data often indicate that feathered lineages were far more ancient than direct fossil evidence initially suggested, pushing back the origins of complex plumage by tens of millions of years, and indicating that feathers were fundamental to dinosaur biology from very early in their evolutionary history. Comparative protein analysis has revealed that dinosaur feathers contained unique structural domains that provided performance advantages not found in modern bird plumage. These molecular innovations apparently allowed prehistoric feathers to maintain function under extreme environmental conditions while providing capabilities that have been lost during subsequent evolutionary refinement for flight optimization. The study of feather arrangement during brooding behaviors has shown that dinosaur parental care rivaled modern birds in sophistication and effectiveness. Fossil specimens preserve precise feather positioning that optimized heat transfer, humidity control, and physical protection for developing eggs and hatchlings. The consistency of these arrangements across multiple species indicates that complex parental care was a fundamental component of dinosaur reproductive success. Chemical fingerprinting of trace elements within individual feathers has revealed the geographic origins of specific specimens, creating chemical maps of dinosaur movements that span entire continents. This approach to studying ancient biogeography provides insights into population connectivity and migration patterns that complement traditional morphological and stratigraphic approaches to understanding dinosaur distribution. The preservation of stress hormone cycles within feather growth bands has provided evidence for the physiological responses that allowed dinosaurs to cope with environmental challenges throughout their lifetimes. Cortisol and related compounds create detailed records of how individual animals responded to drought, predation pressure, social conflicts, and other survival challenges that shaped their daily existence. Biomolecular studies of feather pigment systems have revealed color-producing mechanisms that created visual displays rivaling the most spectacular modern birds. Structural coloration, produced by microscopic feather architectures, would have generated brilliant iridescent effects that served both species recognition and sexual selection functions. The preservation of these nanostructures demonstrates the incredible stability of biological optical systems over geological time. The discovery of antimicrobial compounds within ancient feather structures has shown that dinosaurs possessed sophisticated chemical defense systems protecting their plumage from bacterial and fungal infections. These biochemical shields were apparently more complex than those found in modern birds, suggesting that Mesozoic environments presented unique pathogenic challenges that required enhanced immune responses. The ongoing technological revolution in paleontological research continues to accelerate the pace of discovery, while revealing increasingly sophisticated aspects of dinosaur biology. Each analytical breakthrough opens new research possibilities, while each discovery raises fresh questions about the complexity and diversity of Mesozoic life. The story of feathered dinosaurs has fundamentally transformed our understanding of these remarkable creatures, revealing them as dynamic, behaviorally complex, and often breathtakingly beautiful animals that dominated terrestrial ecosystems for over 100 million years. The revolution in feathered dinosaur research has fundamentally altered how we approach the study of extinct life, transforming paleontology from a discipline focused primarily on bones and teeth into a comprehensive science that can reconstruct behavior, physiology, and ecology with unprecedented precision. When researchers first began identifying feather impressions in Chinese fossils during the 1990s, many scientists were skeptical that such delicate structures could provide reliable information about creatures that lived over 100 million years ago. Today, these same structures are revealing secrets about dinosaur biology that would have seemed impossible to study just a few decades ago. The development of portable analytical equipment has democratized feathered dinosaur research, allowing scientists to conduct sophisticated chemical analysis at at remote field sites where fossils are first discovered. Real-time feedback about preservation quality and specimen composition can now guide excavation strategies, ensuring that the most scientifically valuable specimens receive appropriate treatment from the moment they emerge from the rock. This technological accessibility has enabled discoveries at locations that would have been impossible to study using traditional laboratory-based methods. Artificial intelligence systems trained on vast databases of feather images are revolutionizing how scientists identify and analyze specimens. Machine learning algorithms can process thousands of fossil photographs in the time it would take human experts to examine a handful, while maintaining accuracy rates that often exceed manual identification. These computational tools are systematically revealing feather traces in museum specimens that have been overlooked for decades, effectively expanding the known diversity of feathered species without requiring new fieldwork. The preservation of growth hormones within fossilized feathers has opened entirely new avenues for understanding dinosaur development and life history. Chemical markers associated with rapid growth phases reveal that many species underwent dramatic transformations during specific life stages, with feather structure and function changing in coordination with overall body development. These hormonal signatures provide direct evidence of how dinosaurs balanced the metabolic demands of growth against the energy requirements of maintaining elaborate plumage. Advanced fluid dynamics modeling has revealed that many dinosaur feathers possessed aerodynamic properties that far exceeded the apparent requirements of their lifestyle. Computer simulations show that flightless species often had feathers capable of generating significant lift and thrust, suggesting that the evolutionary machinery for powered flight was in place millions of years before it was actually utilized. This biological over-engineering provides compelling evidence for how complex innovations can arise through gradual modification of existing structures. Chemical analysis of seasonal stress markers preserved in feather growth bands has created detailed annual calendars of dinosaur life, showing how these animals coped with predictable environmental challenges throughout their lifetimes. Signatures associated with food scarcity, temperature extremes, and reproductive stress appear at regular intervals, revealing survival strategies and behavioral adaptations that shaped dinosaur ecology across millions of years of evolutionary time. Isotopic fingerprinting of ancient lipids has reconstructed migration routes that connected dinosaur populations across vast continental distances. Some species regularly traveled between dramatically different climatic zones during annual cycles, covering distances that rivaled modern bird migrations, while navigating landscapes that bore little resemblance to contemporary geography. These chemical pathways through prehistoric terrain reveal ecosystem connectivity that was far more dynamic than previously assumed. Microscopic analysis of trace elements within individual feathers has identified dietary specializations that allowed multiple species to coexist without direct competition for resources. Chemical signatures can pinpoint the specific plant families or invertebrate groups that different dinosaurs preferred, revealing ecological precision that required sophisticated sensory capabilities and foraging behaviors. This level of dietary specialization indicates community structures that approached the complexity of modern tropical ecosystems. The recovery of ancient DNA fragments from exceptionally preserved specimens has begun providing direct genetic evidence of the molecular mechanisms controlling feather development. While complete genomes remain beyond current capabilities, these partial sequences reveal conserved regulatory pathways that have persisted from dinosaurs through modern birds. The genetic toolkit underlying feather biology appears to be remarkably stable across vast spans of evolutionary time. Bacterial communities preserved within fossilized feather follicles have revealed complex microbial ecosystems that played crucial roles in plumage maintenance and coloration. These ancient microorganisms show specialized metabolic pathways that would have contributed to feather health, while potentially producing some of the pigments that created prehistoric color displays. Some bacterial lineages have modern descendants that continue inhabiting bird feathers, representing unbroken evolutionary relationships spanning over 100 million years. Biochemical analysis of fossilized preening oils has shown that dinosaurs produced maintenance compounds of extraordinary complexity, containing waterproofing agents, antimicrobial substances, and ultraviolet protective molecules that exceeded the sophistication of modern bird secretions. The metabolic investment required to synthesize these chemical cocktails emphasizes how critical feather care was to dinosaur survival and reproductive success. Spectroscopic identification of fluorescent compounds has revealed that some dinosaur species possessed communication channels operating in ultraviolet wavelengths, completely invisible to human perception. These hidden signals could convey species identification, reproductive status, and territorial information, while remaining undetectable to predators and competitors lacking appropriate visual systems. The discovery of these cryptic communication networks adds profound complexity to our understanding of dinosaur social behavior. Coordinated molting patterns preserved in multiple specimens show that different body regions underwent carefully timed feather replacement cycles that maintained optimal function throughout the year. Display plumage was typically renewed just before breeding seasons to ensure maximum visual impact during courtship, while structural feathers followed schedules that prioritized thermoregulatory efficiency. This temporal orchestration required hormonal control systems that integrated environmental cues with internal physiological rhythms. Engineering analysis of microscopic feather fasteners has revealed self-repairing mechanisms that could restore structural integrity after mechanical damage without requiring complete replacement. These molecular connectors could re-establish proper alignment even after significant stress, allowing dinosaurs to maintain aerodynamic and insulation properties despite the wear associated with active lifestyles. The repair principles underlying these systems have inspired biomimetic research aimed at developing self-healing composite materials. Protein crystallography studies of ancient keratin have identified structural modifications that provided dinosaur feathers with performance characteristics superior to modern bird plumage in specific applications. These molecular adaptations allowed prehistoric feathers to withstand environmental stresses that would damage contemporary structures, while maintaining flexibility and strength under conditions that no longer exist in current ecosystems. Neural pathway reconstructions from fossilized skulls have provided evidence for brain regions specifically dedicated to complex feather control and display coordination. Endocranial volume dedicated to plumage manipulation correlates directly with feather elaboration across species, indicating that cognitive demands increased alongside morphological sophistication. The neural processing power devoted to feather management suggests that display behaviors required significant mental coordination and behavioral planning. Volcanic ash geochemistry has revealed the precise environmental conditions that created preservation windows for soft tissue fossilization. Optimal conditions occurred when rapid burial combined with specific water chemistry to prevent bacterial decomposition while promoting mineral replacement of organic materials. Understanding these taphonomic processes has guided exploration toward geological formations with similar characteristics, significantly improving discovery success rates. Evidence for communal molting behaviors in fossil assemblages indicates complex social structures that involve group-wide synchronization of feather replacement cycles. These mass molting events required sophisticated communication systems to coordinate timing and location, suggesting social organization that extended far beyond simple reproductive aggregations. The behavioral complexity necessary for these coordinated activities indicates cognitive abilities approaching those of the most socially advanced modern bird species. Biomechanical reconstruction of feather-assisted locomotion has shown that many terrestrial dinosaur species achieved enhanced jumping performance, rapid directional control, and improved stability during high-speed pursuits through coordinated limb movements. Even obligately flightless species possessed aerodynamic capabilities that provided significant advantages during ground-based locomotion, suggesting that the evolutionary transition to powered flight involved multiple intermediate stages serving different functions. Growth stress indicators preserved in feather structure reveal that plumage development was intimately connected to overall health and environmental quality. Poor nutrition or disease resulted in characteristic structural abnormalities that create permanent records of individual survival challenges. These pathological markers provide insight into dinosaur health dynamics and stress responses that would be impossible to study through skeletal remains alone. Acoustic analysis of specialized feather structures has identified sound-producing capabilities that complemented visual displays in dinosaur communication systems. Particular feather arrangements could generate specific frequencies through controlled airflow, enabling long-distance auditory signals that remained effective in dense vegetation where visual communication might be compromised. These acoustic adaptations indicate multimodal sensory systems that integrated visual, auditory, and chemical signaling mechanisms. Seasonal isotopic variations in growth bands have documented annual migration cycles, connecting dinosaur populations across continental scales. Chemical signatures reveal regular travel between dramatically different environmental zones, requiring navigational abilities and physiological adaptations that allowed survival across diverse climatic conditions throughout yearly cycles. These movement patterns indicate ecosystem connectivity that was far more dynamic than static fossil distributions initially suggested. Preserved feather parasites reveal co-evolutionary relationships between dinosaurs and their microscopic associates that persisted across millions of years of shared evolutionary history. Ancient parasites show morphological specializations specifically adapted for exploiting feathered hosts, including attachment structures and feeding mechanisms that mirror those found in modern bird parasites. These intimate ecological relationships represent some of the most detailed interaction networks preserved in the fossil record. Synchrotron radiation analysis continues revealing hidden feather impressions in specimens that have been studied for decades using conventional methods. High-energy imaging can detect structural details that are completely invisible to traditional examination techniques, systematically expanding the known diversity of feathered species through reanalysis of existing museum collections. Every specimen potentially contains undiscovered information waiting to be revealed through advancing analytical capabilities. Molecular clock calculations derived from ancient protein sequences have provided temporal constraints on evolutionary events that often predate the oldest known fossils by significant margins. Biochemical data indicates that feathered lineages were far more ancient than direct morphological evidence initially suggested, pushing back the origins of complex plumage structures and establishing feathers as fundamental components of dinosaur biology from very early in their evolutionary radiation. Comparative protein studies have identified unique structural domains in dinosaur feathers that provided performance advantages not present in modern bird plumage. These molecular innovations enabled prehistoric feathers to maintain function under extreme environmental conditions, while offering capabilities that were subsequently lost during evolutionary optimization for flight-specific requirements. The biochemical solutions developed by dinosaurs represent alternative approaches to feather engineering that were refined for extinct environmental challenges. Brooding behavior preservation has documented dinosaur parental care strategies that matched or exceeded modern birds in sophistication and effectiveness. Fossil specimens capture precise feather positioning that optimized heat transfer, humidity regulation, and physical protection for developing offspring. The consistency of these arrangements across multiple species and geological time periods indicates that complex parental investment was a fundamental component of dinosaur reproductive biology. Trace element fingerprinting within individual feathers has created chemical maps of dinosaur geographic origins and movement patterns that span entire continents. This geochemical approach to studying ancient biogeography provides insights into population connectivity and dispersal routes that complement traditional morphological and stratigraphic methods for understanding species distribution patterns across prehistoric landscapes. Stress hormone preservation within feather growth increments has documented the physiological mechanisms that allowed dinosaurs to survive environmental challenges throughout their individual lifetimes. Cortisol and related compounds create detailed records of how animals responded to drought conditions, predation pressure, social conflicts, and seasonal resource availability that shaped their daily existence and long-term survival strategies. Pigment system analysis has revealed color-producing mechanisms that generated visual displays rivaling the most spectacular modern birds in brilliance and complexity. Structural coloration created by microscopic feather architectures produced iridescent effects that served species recognition and sexual selection functions while demonstrating the incredible optical sophistication achieved through biological evolution. The preservation of these nanostructures across geological timescales represents remarkable stability of complex biological materials. Antimicrobial compound identification within ancient feather structures has documented sophisticated chemical defense systems that protected dinosaur plumage from pathogenic threats. These biochemical protective mechanisms were apparently more elaborate than those found in contemporary birds, suggesting that Mesozoic environments presented unique infectious challenges that required enhanced immunological responses and chemical countermeasures. The technological revolution driving feathered dinosaur research shows no indication of slowing, with each analytical breakthrough revealing increasingly sophisticated aspects of prehistoric biology that challenge our understanding of evolutionary processes and ecological complexity. Every new discovery raises fresh questions about the diversity and behavioral sophistication of Mesozoic life, while demonstrating that dinosaurs were far more remarkable and beautiful than anyone had previously imagined. Modern museum exhibitions worldwide have undergone complete redesigns as accumulated evidence for elaborate dinosaur plumage becomes overwhelming and undeniable. Traditional depictions of uniformly scaly reptilian monsters have been replaced by scientifically accurate reconstructions that showcase the vibrant colors, complex behaviors, and stunning beauty that characterize these remarkable creatures throughout their 165 million year dominance of terrestrial ecosystems. The ongoing research continues expanding our appreciation for the complexity and sophistication of prehistoric life, while demonstrating how evidence-based scientific investigation can completely transform our understanding of natural history. The feathered dinosaur revolution represents more than just a paleontological discovery. It exemplifies how careful observation, technological innovation, and scientific rigor can reveal truths about the natural world that are far more wonderful and surprising than any fiction we could imagine. And there you have it. The next time you walk past that Tyrannosaurus Rex skeleton in a museum, remember that you're not looking at a scaly reptilian monster from some prehistoric nightmare. You're looking at the remains of what was likely a magnificent feathered giant that would have been more spectacular than any creature alive today. The evidence is overwhelming. The science is solid, and the implications are staggering. We've journeyed from that first controversial discovery of Sinosauropteryx in Chinese fossil beds to a complete revolution in how we understand dinosaur biology. These weren't the slow, cold-blooded lizards of old Hollywood movies. They were dynamic, intelligent, behaviorally complex animals with elaborate plumage that rivaled modern birds in beauty and sophistication. They communicated through intricate visual displays, cared for their young with remarkable dedication, and inhabited ecosystems that were far more colourful and vibrant than anyone had ever imagined. The technological advances driving these discoveries continue to accelerate at breakneck speed. Every fossil has the potential to reveal new secrets when subjected to modern analytical techniques. Museums around the world are systematically re-examining their collections, and I guarantee you that more groundbreaking discoveries are just around the corner. This story isn't over. It's just beginning. And if you want to stay on top of the latest developments in paleontology and continue exploring the incredible world of prehistoric life, make sure you're subscribed and hit that notification bell. Because trust me, the next discovery could change everything we think we know about life on Earth. Until next time, keep looking up, keep questioning, and remember that the truth about our planet's past is often far more amazing than anything we could ever imagine.